So hello students, good evening each and every one of you. I hope you're safe and sound and doing extremely well in life. This is Basavaraj, I'm your biology master teacher. So hello Ravindrani, hello Rupa, hello MD, hello Shalini. Good evening each and every one of you. So let me know in the chat, before we start anything, let me know in the chat if my voice is audible to you, if the presentation behind is visible to you. So quickly students in the chat, let me know in the chat right now if you're able to see my voice, if you're able to hear my voice and see my 1 second students, 1 second students, are you able to hear me? 1 second. Mithun. Clear? Is it everything is clear? So can we start today's class? So before we start today's class, I want each and every one of you to like the video. Yes, as usual, mark your attendance by liking the video and sharing the video, and also subscribe to the channel. Yes. So while we wait for everyone to join, let me ask you a simple question. As usual, a simple question as usual. The question for today is it, check the quality once. So the question for today is very simple. The question for today is very simple. Being a dropper or balancing boards with NEET preparation, which is more tougher? Are you in your class 12th right now, or are you a dropper? First, let me know that first of all of you. Let me know in the chat right now if all of you are bored, if you are bored students, or you have taken a drop. So quickly let me know in the chat and we will start today's amazing class. So my favorite topic, yes, it is also my favorite topic, that is anatomy of flowering plants. Quickly let me know in the chat which type of students are you. Students, I'll tell you one thing. Personally, being a dropper and being a board student, both of them have certain difficulties. Will you all of you agree with me? Both the students will have some sort of difficulties. That is, some of the students who are a dropper right now, it can be mentally exhausting. Yes, there is a lot of relatives, right? Your family pressure, from every single side you have pressure. But if you're a class 12 student, you will have problems, that is balancing board, going to college, going to school, writing assignments, writing your exams. Students, but don't worry. Either way, either you are a board student or you are a dropper, you are the same to us. Because we have decided on this channel, we will help you to balance both of them. That is, even if you're a dropper or even if you are a class 12 student, we are here with you. We are here with you. We will make sure that your complete preparation is happening. That is, it could be a detailed class like this, or it could be your notes. We provide you notes, or it could be anything. PDF of your entire session, question paper practice, PYQs, every single thing on this channel. But in return, we are asking only one thing. That is, please be consistent. That is, throughout the session, I want each and every student, one of you, Bhavna, Vinti, Shalini, everyone, each and every MD, Spurti, each and every one of you, please stay till the end. Be consistent. That is the only thing we are asking from you. So can we start? Yes. Can we start the class? Promise me. Each and every one of you, promise me right now that today you will confirm those 16 to 14 marks today. Yes. Sir, this chapter is a trauma. Hope you will clear. I will clear each and every word to you today. So students, I hope you will stay till the end. Not like these fake students who will stay in the starting and leave the end. Promise me right now, students, you will stay with me towards the end of the session and you will tell in the end to me, yes sir, I have confirmed the 14 marks or 16 marks in this class by attending today's lecture. Amazing. I feel all of you will stay till the end. So I am keeping your promise. I am taking your promise and we'll start today's class. Yes. So students, anatomy of flowering plants. So what is the meaning of anatomy? So students, I want each and every one of you to hit the like button right now. And everyone, each and every one of you, green hearts in the chat. And I will teach you morphology, which I taught you before. Even anatomy today's class. Every single word from your NCERT will be covered. Every single PYQ, which is very important, will be covered in today's video. So without wasting any time, let me teach you what is meaning of your anatomy. Students, In morphology, we learnt about the external forms of feature. Yes, the entire external we learnt. But in the case of your anatomy, we will be learning the internal structure, how the plant looks from the inside. Generally, students, every plant from the inside is uniform. Listen to my words very carefully. In general, each and every plant from the inside is always the same. But in some cases, there might be certain variations. Yes, there might be some certain variations. For example, internally, every single plant has an epidermis. Every single tissue has an epidermis, a cortex, and a stele. But in certain plants, such as your adaptations, it could be your hydrarch or xerarch, right? Hydrophytic plants, xerophytic plants. In certain plants like this, you can find certain modifications. But internally, each and every plant is always the same. So students, let me start by a simple topic today. That is your plant tissues. So everyone in the chat should tell me what is a tissue. All of us know. So students, write down every single one of you, please write down with me. That is the only thing I'll ask you. Keep your NCERT next to you. Keep my lecture in front of you. Keep your notebook towards your right side. Keep writing the notes. With me. Follow the NCERT with me, and you will confirm the marks today. So what is a tissue? Tissue is nothing but a group of cells. Yes, tissue is nothing but a group of cells. Tissue is nothing but a group of cells. Now students, I'll ask you one thing here. This group of cells, they can have a same origin. Yes, all of the cells here can have a same origin. Yes, they can have same origin. Now all the cells here can have a same function. Yes, when a group of cells come together, we call it a tissue. Now this particular tissue can have same origin and same function, but do they have same structure? When you were in your class 9th, in your class 10th, we learned that every single cell or every single tissue is same in structure. But is it true? The answer is actually no. No is the answer. That is, the particular type of tissue, any tissue can be same in structure, but a tissue can also be different in structure. So write down with me, a particular tissue can be same or different in structure. Now the example for same is your parenchyma. Every single cell in the case of your parenchyma is always the same. But in the case of your different, that is your xylem and phloem. In case of your xylem and phloem, every single cell or tissue can vary. So what is this? What is a tissue? Tissue is nothing but a group of cells which have a same origin, which have a same function, but structurally they differ. Sometimes the structure can be same in the case of your parenchyma, or the structure can become different. That in the case of your xylem and phloem. Clear? The next point I want all of you to understand is types of tissues. Next thing I want all of you to understand is the types of tissues. What are the different types of tissues, sir? Simple classification, I'll tell you. Very simple classification. In types of tissues is mainly divided into two types. The first type is called as your meristematic cells. First one is called as your meristematic cells. Meristematic cells or meristematic tissue. The second one is called as your permanent tissue. Second one is called as what? Permanent tissue. Now, how are they different? Listen to me very carefully now. Your meristematic tissue is always dividing. Yes, always dividing. Listen to me very carefully. This meristematic cells is always dividing. They have something called as dividing cells. And remember this very carefully. The meristematic cells are living cells. They're what? They're living cells. Now, what about your permanent tissue? Your permanent tissue is not dividing, but it has a very specific function. Your meristematic cells decides one day, no, I do not want to divide anymore. I will just become permanent. I'll become permanent and I will do a very specific function. That is only one function, that is your meristematic cells as well as your permanent cells. So your permanent cells will have a very specific function. Now, next point here is this permanent cell can be living or dead. They can be living or dead. And this particular permanent cell can be divided into two types again. That permanent cell can be divided into two types, that is your simple tissue or complex tissue. So what do we have? In types of tissue, we have two types. Tissue, we have two types. The first one is called as your meristematic tissue, which is a dividing cells, which are living cells. Apart from that, we have your permanent cells. Permanent cells do not divide. Yes, they become permanent and they have a very specific function. They are living or dead. They can be living or dead. And if you further divide your living or dead tissues, permanent tissues, we get simple tissues as well as complex tissues. Till now, does anyone has any doubt? Ask me right now. The next 30 seconds, I will take all the doubts. Next 30 seconds, I'll take all the doubts fast. If you have any doubt, ask me right now. Any doubt till here? If not, I will teach you the next part, that is your meristematic tissue. Now, I told you, meristematic tissues are nothing but your dividing cells. Yes, meristematic cells are dividing cells. Now, please write down with me certain properties of your meristematic cells. Will you? Please write down with me certain properties of your meristematic cells. The first property here is all of these cells are very small cells. They're very small cells because they are dividing. And if cell is dividing, does it have dense cytoplasm or very less cytoplasm? The answer is very simple. You remember, every single time a cell divides, it needs nutrition. It divides your cytoplasm. So in the case of your meristematic cells, the cytoplasm is very dense, very compact, very dense cytoplasm. Now, what about intercellular spaces? In the case of your meristematic cells, the intercellular space is actually absent. Clear? The next point here is, since it is very dividing cells, since these are dividing cells, the cell wall here is actually very thin cell wall, which has very less depositions or no depositions at all. Very thin cells, and it has a primary cell. So write down with me, the cells here are very thin-walled cells, which are arranged in a very compact manner. They're arranged. And the cell wall here, remember always, if the cell wall is thin, if the cell wall is actually very thin, it is always a primary cell wall. Primary cell wall. Now, what about nucleus? Can anyone in the chat tell me what about the nucleus? Will it have a very tiny nucleus or very large conspicuous nucleus? Remember, students, I will show you a diagram now, microscopic image now, to show you how the nucleus looks like. Are you ready for that? Are you ready for a microscopic image? Tell me fast. The nucleus here is very large. It have a very large nucleus. Large nucleus. Very large nucleus. If the nucleus is very large, the vacuole is usually pushed outside. The vacuole is always pushed outside. So, can I write vacuole is actually absent here? Vacuole is absent here. So, what are the properties of your meristematic cells? The properties of your meristematic cells is your small cells. They have a very dense cytoplasm for dividing. Then we have intercellular spaces is absent. Why? Because they are very compactly arranged. They have very thin walled cells. The cells here are primary cell wall. Then we have very large nucleus. Then we have vacuoles are actually completely absent here. The vacuoles are completely absent here. Look at the diagram here. Is the diagram visible to all of you? Is the diagram visible to all of you? If you look the cells here, these are the dividing cells. See, can you see the different cell stages? Yes, different cell stages. We have very thin walled cells, very large nucleus. Is there any intercellular space? Intercellular space is completely absent. That each and every cell is very compactly arranged here. Very compactly arranged, and they have something called as dense cytoplasm. That is the microscopic image. So what are the properties? Let me tell you once again, please remember them. That is small cells, dense cytoplasm, intercellular space is completely absent, thin walled cells, primary cell wall, large nucleus, and vacuole is pushed outside. There is complete absence of your vacuoles. Clear? Any doubt in the properties? Tell me first. Any doubt in the properties of your meristematic cells or meristematic tissue? This is the meristematic tissue here. Now, if you have understood, if you have some idea about meristematic tissue, can I classify it now? Let's try to classify, let's try to segregate them. Let's try to separate them out. Can we? Let's classify. Ready? All of you? Now we'll do classification of your meristem. Now, your NCERT doesn't tell very clearly, but there are two types of classification. One type of your classification is based on the origin or based on the growth. So based on the origin, your meristematic tissue can be divided into two types. The first one is your primary meristem. The other one is your secondary meristem. Now, sir, what is the difference between primary and secondary? Now, you tell me. First class to fifth class, you were in primary section. That was your starting stage of your schooling. Then you went to secondary class. Right? Then you went to secondary class. When you went to secondary class, that time you were a little grown. Your height was increased. Right? Your height was increased. Your mind also was matured. Just like that, even your meristem, even your case of meristem, which is classified based on the origin, we have primary meristem and secondary meristem. Primary meristem is present from beginning. It is present from beginning. All the way from the starting stage of the plant's life. From the starting stage of the plant's life, this meristem is present in the plant. This meristem is present in the plant. Now, what about secondary meristem? Just like how you went from primary school to secondary school, your plant also went from primary to secondary. So, secondary stage happens after secondary meristem is formed later on. It is formed later on in the plant's life. So, in the starting of the plant life, we have the primary meristem, but later on, after few years, in the plant's life, we have the secondary meristem that is formed later on. Clear? Now, I'll tell you one more point here. Your primary meristem is responsible for the primary growth. Primary meristem is responsible for the primary growth. Now, what is this primary growth? Primary growth is nothing but increase in height. Yes, that's how you did, right? When you were in small classes, Your knowledge increased very fast. All of you, trust me, in your report card, if you go check your primary class report card, all of you would be getting A plus, right? A plus in science, A plus in social, A plus in English, A plus in Hindi, right? All of you would are getting. That is a sudden increase in knowledge. Just like in your plants, in the primary meristrum. In the primary meristrum, primary growth happens. What is the primary growth? Primary growth is nothing but, that is, increase in height of the plant. Increase in height of the plant is your primary growth. Then we have your secondary growth. In secondary growth, what happens? In your secondary meristrum, we have secondary growth. In your secondary meristrum, we have the secondary growth. In secondary growth, what happens? Your width of the plant increases. Width or girth. Width or girth of the plant. It increases like this. Starting it increased like this. Nicely it increased. After some years, it started increasing like this. That is called as your, because of your secondary meristrum. Secondary meristrum. Now your primary meristrum. Primary meristrum happens in your both monocot and dicot both. Listen to me very carefully. In case of your primary meristrum, it happens in both. That is, it happens in the case of your monocot as well as in the case of your dicot. But secondary growth does not happen in the case of your monocot. Secondary growth or your secondary meristrum is only found in your dicots. It is only found in your dicots. Only in dicots. So, if you want to classify the meristrum based on the origin, we have your primary meristrum, we have the primary meristrum, and we have the secondary meristrum. Primary meristrum is from the beginning, right? From the beginning of the plant's life, primary meristrum is always there. Then secondary meristrum in the plant's life comes later on. It comes later on. Your primary meristrum is responsible for the primary growth. What is primary growth? Increase in the height of the plant. While your secondary growth happens in the case of secondary meristrum, that is increase in width or girth. And this secondary meristrum or secondary growth is only and only seen in the case of your dicots. But your primary meristrum is seen in the case of both monocot and dicot. Is the point clear to all of you? If this is clear, can we classify the plants based on the location now? Can we classify? Quickly tell me in the chat. Quickly tell me in the chat, my dear future doctors. Can we classify your meristematic tissue now based on the location where it is found? Okay. Now based on your location now. Based on the location. Like how you have your house, I have my house. My house in first floor, your house is in ground floor. That is different location. I am in Bangalore. Tell me where in the chat right now fast. Because I'm in Bangalore. Jai Singh is probably from, let's guess, Rajasthan. Then we have your Shivani. Probably Shivani is from Bangalore or Chennai or Tamil Nadu or Andhra Pradesh. You never know. So that is based on your location. Based on your location, we have three types. Based on location, your meristematic tissue is divided into three types. What are the three types? The first one is your apical meristrum. The first one is your apical meristrum. Amazing. The first one is your apical meristrum. The second one is your intercalary meristrum. Intercalary meristrum. The third one is called as your lateral meristrum. Write down with me, all of you. Lateral meristrum. So based on the location, we have your apical meristrum, intercalary meristrum, and lateral meristrum. Now, tell me, where do you find your apical meristrum? The meaning of apical. The meaning of apical, apex. Apex or apical means top or tip. So it is called as what? Tip. So it is found in your apex region. Apex or tip region. Tip region of what? Tip region of your fingernails. Do you find your apical meristrum here? No. Where do you find it? In the tip regions of your growing root or growing shoot. So can I write it is found at the top or the apex or top of what? Of your shoot apical meristrum and root apical meristrum. SAM or RAM. SAM is the shoot apical meristrum, which is the growing tip of the shoot. Or we have RAM. RAM is what? Growing root tip. So where do you find where do you find your apical meristrum? Apical meristrum is found where? In the shoot apical meristrum or root apical meristrum. Now, apical meristrum is actually a type of your primary meristrum. It is a type of your primary meristrum. Why, sir? How can you tell me that? I told you, students, primary meristrum is responsible for the height of the plant. Now you tell me in the chat, does the SAM increase the height of the plant? Yes, sir. Does the RAM increase the underground length of the root? Yes, sir. So your apical meristrum is a type of your primary meristrum. Next one, come learn with me. Your intercalary meristrum. Your intercalary meristrum is actually found. It is actually found between. It is actually, this is a meristrum. Meristrum found between your permanent tissue. Between your permanent tissue or mature tissue. Or mature tissue. Don't worry if you didn't understand this line. I will show you diagrammatically. Then you will understand this better. You're like, oh sir, yes sir, that is only correct. Don't worry if you don't understand this line. I will come back to this line and I'll make sure you understand. Okay. Now, next point here is lateral meristrum. Lateral meristrum is present on the sides. It is present on sides. Now, what do I mean by sides? I will show you diagrammatically, then you will understand what is the meaning of present on the sides. Now, before I go to the diagram, let me tell you where it is present also. Now, this particular apical meristrum is found in your monocots and dicots both. But this intercalary meristrum is also a type of your primary meristrum. And what is the function here? The function of your intercalary meristrum is it increases the internode. It increases the internode. What about lateral meristrum? Lateral meristrum provides it is a type of your secondary meristrum and it is found only in dicots. Now listen to me very carefully. Listen to me very, very carefully, all of you now. Apical meristrum is found at the shoot tips. Yes, shoot tips and root tips. Where? On the SAM or RAM. Shoot apical meristrum or root apical meristrum. Then it is a type of your primary meristrum. Clear? Now your intercalary meristrum. I told you intercalary meristrum is a meristrum found between permanent tissue or mature tissue. What is the meaning of that? Now if you look at the diagram here, this is your apical meristrum. Yes, growing tip. Now intercalary meristrum is found at the nodal regions. See this is a node. This is a node. At this nodal region, we find the intercalary meristrum between the two permanent cells. Between the two permanent cells or permanent tissues, intercalary meristrum is present. Between two permanent tissue, intercalary meristrum is present at the nodal regions. What does it do? What is the function? The function of your intercalary meristrum, which is found only in your monocots, what does it do? It increases the internode length. So if this is present here, if your intercalary meristrum is present at the nodal region, it will increase the length of the internode. It will increase the length of internode. This is the node, this is a node. Between these two, internode, it will increase. That is why, in the case of your grasses, in the case of your sugarcane, have you eaten sugarcane? Sankranti, recently you ate, na? Recently you ate sugarcane. All of you. Sugarcane. In that sugarcane, you might have seen there were small gaps. This in the sugarcane when you eat, there is a soft part which you eat. Then there is one hard part here somewhere in the middle. This is the nodal region. Between the two nodes, we have the internode. In that internode, it increases in length. Why? Because of your intercalary meristrum. Now, what about lateral meristrum? Lateral meristrum is present on the sides of the stem. If this is the main stem, it is present on the sides. What does it do? It increases the girth, just like your secondary meristrum. Your lateral meristrum is responsible for increasing girth. That is fat trees you have, no? Woody fat trees. Why? Because of your lateral meristrum. Because of lateral meristrum, we have the thick woody trees. So based on location, we have what? Apical meristrum, shoot tips, intercalary meristrum present between the two permanent cells at the nodal region, increases the length of the internode. Then we have lateral meristrum. What does lateral meristrum do? Lateral meristrum increases the girth. Lateral meristrum increases the girth of the plant. Clear? Sir, in case of sugarcane, yes, David. In the case of your sugarcane also, your intercalary meristrum increases the internode. Now, someone was asking me a question here. The question was, sir, can you quickly tell me difference between primary and secondary meristrum? Don't worry, students. All of these notes will be provided to you on the Telegram channel. Primary meristrum, responsible primary growth, told you already. Increase in root and shoot length, already told you. Apical meristrum is a type of your primary meristrum, already told you. Present in root and shoot apices, already mentioned. What does it give you? Primary meristrum provides primary tissues. Primary meristrum gives us primary tissues. Primary permanent tissues. Primary permanent tissue. Now, one famous question is there, all of you. Sir, from primary meristrum, do we get secondary meristrum? I know all of you, this question might be there in your head. Everyone might be thinking, from primary meristrum, we get secondary meristrum. Some of you think that? Do some of you have the question in your head? If you have the question in your head, it will be cleared right now. No. Primary meristrum doesn't give us secondary meristrum. In fact, in primary meristrum, we have your primary tissue, right? Primary permanent tissue. This primary permanent tissue de-differentiates. This primary permanent tissue de-differentiates and becomes the secondary meristrum. It is the secondary meristrum. So, your primary meristrum is not giving you secondary meristrum. That confusion should be cleared today only. Primary meristrum is not giving you secondary meristrum, but in fact, primary permanent tissue is undergoing de-differentiation. Is undergoing de-differentiation to provide you secondary meristrum. Is that concept clear to all of you? Sir, but sir, primary, but after primary only secondary comes. It comes after secondary. But does the primary gets converted to secondary? Answer is not. Primary meristrum doesn't get converted to secondary meristrum. Primary meristrum will give you primary permanent tissue. That primary permanent tissue will de-differentiate and that will become your secondary meristrum. Clear? Do you want me to repeat that concept? Because I don't want my students to by heart all this. I want my students to understand the botany, enjoy the botany, understand how things happen. Okay? Amazing all of you are. I'll give you one more example. You have one meristematic cell. You have one meristematic cell. Now this meristematic cell, right? This meristematic cell will lose its ability to divide. Lose its ability to divide. It becomes primary permanent cell. Meristematic cell losing its ability to divide, it is becoming primary permanent cell. That this meristematic cell can be anything. Now this can become your medullary race. Example for your primary permanent cell is your medullary race. race. Medullary race is an example for your primary permanent cell. Now this primary permanent cell can undergo dedifferentiation, right? It can undergo dedifferentiation. Can undergo dedifferentiation. If this primary permanent cell undergoes dedifferentiation, it gives us the secondary meristrum. That is what I told you here. This primary tissue here, primary permanent cell is undergoing dedifferentiation and we are getting the secondary meristrum. For example, your medullary race. Medullary race is a type of your primary permanent cell. Now this medullary race can undergo dedifferentiation. This medullary race can undergo dedifferentiation and become your vascular cambium. Can become vascular cambium. Vascular cambium is a type of your secondary meristrum. Understanding? If you're not understanding, please ask me right now what is dedifferentiation. I'll teach you again, don't worry. If you do not know this concept, I'll teach you one more time today. I'll teach this entire concept today one more time. If you do not know what is dedifferentiation, redifferentiation, what is differentiation at all, stay with me today, watch the video till the end and I'll tell you. Sir, I think primary converts to secondary. No, primary doesn't get, never. Primary meristrum will never convert to secondary meristrum. Your primary meristrum will give you primary tissue. Primary meristrum will give you primary permanent tissue. That permanent tissue will become secondary meristrum. It is never always like this. It is always like this. Clear? Can we tell you one diagram? Can I tell you one amazing diagram here? This amazing diagram is from your NCERT. This amazing diagram is from your NCERT. Understand with me, all of you? Now here, let me show you. This is your root apical meristrum. Yes, sir. This dome-shaped structure. This dome-shaped structure is your root apical meristrum, also called as your RAM. RAM. Root apical meristrum. Now this root apical meristrum will divide like this. It will also divide like this. Root apical meristrum will divide on both the sides. When the root apical meristrum divides this way, it gives us the initials of root cap. When the root apical meristrum divides this side, it gives us the initials of root cap. Now this initials of root cap later becomes the root cap. So you have your root apical meristrum which divides on both the sides, which provides us initials of root cap initially when divides, and later on initials of root cap will convert to your root cap. Now what do we have on the upper side? On the upper side we have your initials of your central cylinder and cortex. Central cylinder and cortex. Now this central cylinder later gets converted to your vascular bundles. See here, this central cylinder is nothing but your vascular bundles. If you look at the section, of your root, take one section of your root. The section of root will have epidermis, that is called as epiblema. Then we have the middle cortex, that is nothing but your central cylinder. Right, so we have your cortex, then we have your vascular bundles, right? So this protoderm, can you see? This protoderm is nothing but your epidermis. The protoderm here gets converted to your epidermis, cortex becomes cortex, central cylinder becomes your vascular bundle. Let me recap one more time for all of you. We have your root apical meristrum here, which divides on both the sides. When it divides this side, it provides us initials of root cap. Now this initials of root cap gets converted to root cap. Done, sir. When the RAM divides on the upper side, it gives us your initials. What is the meaning of initials? Initials means starting cells. Initials means starting cells. Initials of your central cylinder and cortex. Now then we have your protoderm. Protoderm becomes the epidermis later on of your root, that is called as epiblema. Then we have the cortex of your root, that becomes the cortex. Then we have central cylinder, that is nothing but your vascular bundles. Diagram decoded completely, just like that. Then we have your shoot apical meristrum here, also called as your SAM. Now this particular SAM will divide on both the sides. Yes, the SAM will divide here. The SAM will divide on this side and on this side. When the SAM divides on the lower side, it gives us the differentiated vascular tissues. It gives us differentiated vascular tissues. If your SAM divides on the upper side, when the SAM, also called as your SAM, when the SAM is going upwards, when the SAM is growing upwards, what is it producing? When the SAM is growing, it is producing leaves. If you know this diagram, students, if you remember the diagram which I taught you, this is your stem, this is your stem, this is your apical meristrum, also called as your shoot apical meristrum. In morphology class I mentioned, as the SAM is growing, upwards, it is producing leaves in an acropatal manner. As the SAM is growing upwards, it is forming leaves. It is producing leaves. It is producing what? It is producing leaves. And when as the SAM is growing up and we are getting leaves, some of the cells are left behind. As the SAM is growing, some cells are left behind. Where some cells are left behind in the leaf axil. This is called as leaf axil. As the SAM is growing, some cells are left behind in the leaf axil. That some cells left behind later gets converted to your axillary bud. Gets converted to your axillary bud. Now this axillary bud can produce your branches or flower. So as the SAM is growing, as the shoot apical meristrum is growing up, the leaves are forming in an acropatal manner, and as the leaves are forming, some cells are left behind. Some cells are left behind in the leaf axil. That cell later gets converted to your axillary bud, and that axillary bud later gets converted to this axillary bud here. Axillary bud can become what? A branch or a flower. This axillary bud right here can become a branch or a flower. Clear? Is it clear? Do you know what is SAM now? You know what is shoot apical meristrum? This is a line from your NCERT. Now I'll make you read NCERT and you will understand. So someone is saying, sir, this is not in your new NCERT. Yes, it is not there in new NCERT. But you have to study this from your old NCERT. Why? Because this is in your syllabus for your NEET 2024. It is in your syllabus. Clear? So as the SAM is growing up, leaf primordia are formed. What is leaf primordia? Leaf primordia is nothing but initials of your leaves. As the SAM is growing up, leaves are developed on the acropatal succession. And as the leaves are developing, some cells are left behind in the leaf axil that later gets converted to your axillary bud. Clear, all of you? In the chat, I'll make you read NCERT now. Can we read NCERT one by one line, students? This is a complete packed session. I have detailed lecture. I have NCERT snips for you. Then I also have PYQs for you. So can we start? See here, read with me. Meristomal tissue growth in plants is largely restricted to your specialized regions of actively cell division. That is called as your meristrum. Done. Plants have different kinds of meristrum. Yes, based on origin, based on location, right? Meristrum which occur at the tips of your root and shoot and produces your primary tissue are called as apical meristrum. Yes, we discussed already. We know what is apical meristrum. Apical meristrum is a meristrum which is found in your tips of your shoot and root. Yes. Then we have what? Then we have root apical meristrum occupies the tip of the root. Yes, RAM is at the tip of the root. Then we have the shoot apical meristrum occupies the distant most position region of your stem axis. That is your SAM is at the shoot, ram is at the root tip. During the formation of leaves, elongation of the stem, some cells are left behind. I told you, as the SAM is growing, some cells are left behind. See, formation of leaves and elongation of the stem, some cells are left behind from the shoot apical meristrum. What is shoot apical meristrum? As the SAM is growing, some cells are left behind, constitute the axillary bud. That's how you get the axillary bud. Okay, such buds are present in the axil of the leaf. Where are they present? In the leaf axil. I drew the diagram. And are capable of forming the branch or flower. They can make a branch or they can make a flower. The meristrum which occurs between the mature tissue. I told you, the meristrum which is present between the tissue, that is called as your intercalary meristrum. See, intercalary meristrum. They occur in grasses, yes, in monocots, and also sugarcane, I told you, and regenerate the paths removed by the grazing herbivores. As the herbivores are eating the grass, internode elongation happens again because of the intercalary meristrum. Now both apical meristrum and intercalary meristrum are primary meristrum. I made you write down in the graph, so not graph, in the table, intercalary meristrum and apical meristrum are primary meristrum because they appear where? See, appear early in life of the plant. That point also we mentioned. Primary meristrum is a meristrum which is found in the early life of the plant and contribute to the formation of your primary plant body, that is increase in the height of the plant. See, every line of NCERT is covered. Every line I'm teaching you today, every line I'm fixing in your brain today. The meristrum that occurs in the mature regions of the root and shoot, many plants, particularly those that produce the woody axis. Here they are telling you about your secondary growth, that is present meristrum that occurs in the mature regions, later stages in plant's life, and shoot if particularly those produce your woody axis and appear later than the primary meristrum is called as your secondary meristrum, also called as your lateral meristrum, Clear? Then we have their cylindrical meristrum. We have example here. Listen to me very carefully. We have examples here. Fascicular vascular cambium. That is the first example of your lateral meristrum. Interfascicular cambium. Second one. Third one is your cork cambium. These three are examples of your lateral meristrum, not the secondary meristrum. That are the golden words. Why, sir? Because students, listen to me very carefully now. NCERT here is very confusing. If you are in my NCERT line by line, I told you hidden line here. There is a hidden mistake here. What is the hidden mistake? NCERT is telling you, very smartly, NCERT is telling you, secondary meristrum and lateral meristrum are same. Yes, NCERT is telling you, secondary meristrum and lateral meristrum are same. So meaning of that is fascicular cambium, fascicular vascular cambium, interfascicular cambium and cork cambium are these three type of your tissues secondary in origin. I told you fascicular vascular cambium, interfascicular cambium and cork cambium are examples of your lateral meristrum. Yes, sir, all three of them are examples of your lateral meristrum. But are all three of them your secondary meristrum? The answer is actually no. The answer is no for this. All three of them are not secondary meristrum. In fact, listen to me very carefully. Your fascicular vascular cambium is also called as your intrafascicular cambium. Intrafascicular cambium. This intrafascicular cambium is primary in origin. It is a type of primary meristrum. While your interfascicular cambium and cork cambium are secondary meristrum. Clear? Is that point in your NCERT? Write down today. Trust me, this is a mistake where so many students might make. These are the mistakes which NCERT also has given. So please rectify this and understand this. Your fascicular cambium, which is also called as your intrafascicular cambium, is primary meristrum, while your interfascicular cambium and cork cambium are secondary in origin. They are what? Secondary in origin. Clear? Is that point clear to all my future doctors? If that point is clear, I want all of you to write down also. I want all of you to write down that point. Let me tell you one more thing here. This is your primary in origin, secondary in origin. This is secondary in origin. Now, if this is your normal cell, yes, inside the tissue or cell we have your cambium like this. We have the cambium present like this. Here we have xylem phloem, xylem phloem, xylem phloem, xylem phloem, xylem and phloem. Now listen to me very carefully. Listen to me very, very carefully. Here we have certain type of connection. Here in the middle we have one more connection. Let me do it green. Now label this very carefully. Now here, this green color one, which is present between your xylem and phloem. The green color cambium, which is present between your xylem and phloem, is called as your fascicular vascular cambium. Also called as intra. Intra means within. Intra classes. Intra-class means within a class. Inter-class means between the classes. Inter is interval. Interval you have in your class, right? Interval means what? Break or between. Right? So you have intra. This is your intrafascicular cambium. Intrafascicular cambium. Intrafascicular cambium or your fascicular vascular cambium. That is present between your xylem and phloem. It is present between your xylem and phloem. Now what about this red one, sir? What about this red one? This red one is nothing but your interfascicular cambium. It is what? Interfascicular cambium. It is interfascicular cambium. So what is interfascicular cambium is present between the two vascular bundles. Interfascicular cambium is present between the two vascular bundles. But your intra or fascicular vascular cambium is present between your xylem and phloem. So your intra is present where? Between xylem and phloem. Inter is present where? Inter is present between two different vascular bundles. This was your NEET previous year question. This was a NEET previous year question. Okay? Clear with all of you? Any doubt till now? Please ask me right now. Please ask me right now. Now this lateral meristrum, fascicular cambium, interfascicular cambium and cork cambium are examples of your lateral meristrum. This is also a NEET PYQ. These are responsible for producing the secondary tissue. They are responsible for the secondary tissue. Inter is present between the two vascular bundles, while your intra is present between xylem and phloem. Amazing. All of you are smart. All of you are very, very smart kids. Now, following the division of the cells in both primary and as well as secondary meristrum, the newly formed cells become structurally and functionally specialized, lose its ability to divide. Such cells are called as permanent or mature cells. Permanent and mature cells are the cells which lose its ability to divide and they become permanent cell. They can be living or dead. They have a very specific function, not dividing but. Okay. The specific regions of apical meristrum produces your dermal tissues, ground tissues and vascular tissues. See, this is your lateral meristrum. Lateral meristrum is of three types: intrafascicular cambium, interfascicular cambium, as well as cork cambium. Write down this. Write down this point. It is intrafascicular cambium, interfascicular cambium, or cork cambium. Amazing. Right? Can we solve a question? Can all my students, all my future doctors solve this question? It's a NEET 2022 question. NEET 22 question. Axillary bud are derived from the activity of lateral meristrum, secondary meristrum, apical meristrum, or intercalary meristrum. Fast tell me. Answer is nothing but your 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. The answer is nothing but your apical meristrum. Apical meristrum is nothing but your SAM. Shoot apical meristrum. As the shoot apical meristrum is growing, some cells are left behind. That becomes your axillary bud. Amazing. It is not intercalary meristrum. Intercalary meristrum is responsible for the secondary, responsible for your what? Internode elongation. Intercalary meristrum is responsible for your internode elongation. Okay. Next question. Next question here. Can all of you see the question? All of you, my future doctors? The question here is, consider, last year question students, last year question this is. Consider the following plant tissues. Yes, we have certain plant tissues. Axillary bud, fascicular, vascular cambium, interfascicular cambium, cork cambium, intercalary meristrum. 4 to 5 different tissues. Identify the lateral meristrum among them. Which one of the following or how many of them are your lateral meristrum? Quickly. 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. That is your intercalary. This is there. All three are there. No? B, C, and D here. Second is the option. Second is the clear. Understanding. Are you understanding the NEET level questions now? How the questions will be asked in your examination? Are you able to solve these questions? Students, if you are able to solve such questions, try solving this question also. What is the question here? Interfascicular. Inter, not intra. Interfascicular cambium is present between primary xylem and primary phloem, pericycle and endodermis, two vascular bundles, or secondary xylem and secondary phloem. 10 seconds, solve the question. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. The answer is between the two vascular bundles. I told you, remember, between the two vascular bundles we have the inter. Between xylem and phloem we have the intra. Amazing. This is a question from your NEET 2022 question. Between the xylem and the phloem, that is intra. Between the two vascular bundles, it is your interfascicular cambium. Are my students able to solve such previous year NEET questions with such ease? Yes, easy questions, very easy chapter. Now, can we start with the permanent tissue? Can we start with your permanent tissues, all of you? Students, if you have understood till now, deliberately I am teaching you very slow. I am taking it slow, that every step you understand my teaching, you understand NCERT, and you need to solve previous year questions. This is the pattern I want all of you to also follow. Watch my session, read NCERT, solve questions. You are set. Concept is clear. Applying the concept, reading NCERT, everything is clear. Right? Everything is clear. Amazing. All of you are amazing. Now, let's start your permanent tissues now. Let's start your permanent tissue. Now, permanent tissue. Now, before that, okay, never mind. I'll teach you differentiation, dedifferentiation, redifferentiation. I've already taught you in the plant growth regulators, but in the end, I'll teach you once again. Plant growth, all of that. But let's understand permanent tissue now. Now students, listen to me very carefully. Permanent tissue are a type of tissue which has a permanent government job, right? It has a permanent government job, So this particular permanent tissue has a very specific function. Yes, very specific function. Do they divide? No sir, they don't divide. They don't divide at all. They are not dividing. Permanent, very specific function. And if I were to divide, if I divide your permanent tissue into two types, permanent tissues can be divided into two types. The first one is your simple permanent. The first one is your simple permanent. Very simple. Simple sada. Simple permanent. Then we also have your complex permanent. Right? Very complex they are. Complex permanent. Simple permanent as well as complex permanent. Okay. Now this simple. What is the meaning of simple? Why are they simple? Because they only have one type of cell. They only have your one type of cell. Only one type of cell. Now what about your complex permanent cell? Complex permanent cell can have more than one cell. They can have more than one type of cell. They can have more than one type of cell. More than one type of cell. Let me tell you some examples. Your simple permanent students. Your simple permanent example is nothing but your simple permanent is your parenchyma. Yes, all of us know. Parenchyma, collenchyma, and sclerenchyma. Collenchyma as well as sclerenchyma. Parenchyma, collenchyma, and sclerenchyma. But your complex permanent are examples are your xylem and phloem. They're what? Xylem and phloem. Read with me. They're xylem and phloem. Xylem and phloem. Xylem and phloem. Clear? Then if I don't write full thing, you'll say, sir, you're not writing full. Now, let's focus on one by one. Let's focus one by one on your simple permanent now. Let's focus one by one on your simple permanent cell. The first one which is given in your NCERT is nothing but your parenchyma. Then we have collenchyma. Then we have your sclerenchyma cells. Parenchyma, collenchyma, sclerenchyma. Now some of the students will be asking, sir, this is not there in new NCERT. But students, do you need to study this? Do you need to study this, students? The answer is yes, Madhumita, you need to study this. Even if it is not there in your new NCERT, it is there in your NEET syllabus. Okay, it is there in your NEET syllabus. Now, let's start with your simple permanent now. Let's start with your simple permanent. The first one is your parenchyma. So students, write down with me all the different properties first. All the different properties. The first property here. The first property of your parenchyma cells is it is like a very common tissue. It is a very, very common tissue. It is a very common tissue. Simple. It is a common tissue. Very common tissue. What is it used for? The main purpose of your parenchyma is packing. P for parenchyma, P for packing. You use it for different types of packaging. Different type of packaging. Yes. Now, here we get interesting. This is where we get interesting. This parenchyma is present in majority of the organs, in all the organs of your plant. It could be your epidermis, cortex. The major of the tissues, major of the organs is actually made up of your parenchyma. So it is a major component. It is a major component of all organs, here, of all organs. The cells here are very thin walled cells. The cells here are very thin walled cells, very thin walled. Now, do they have intercellular spaces? If you ask me, If you open your NCERT, can you see this interstellar space here? In your NCERT also it mentions it has interstellar space, but interstellar space is very, very less. Interstellar space is very, very less. So if I talk about interstellar space, that is ICS, is very less or absent in some cases. Now what is the shape? What is the shape of this particular type of cell? The shape here is very different. Not different, it is actually same. The shape here is called as isodiametric cells. What is the meaning of iso? Iso means same. Diameter means you know the diameter of a particular oval shaped cell. Here this is isodiametric, means the diameter is same on all the four sides. So what is the meaning of isodiametric? The meaning of isodiametric is the diameter is same on all the four sides. Same diameter, right? But do they also have various shapes? Yes, sir. They come in various shapes. Various shapes such as your spherical. Yes, some can be your spherical in shape, some can be your oval in shape, some can be round in shape. Yes, they can be round, they can be polygonal also. Yes, they can be polygonal. They can also be elongated. They can also be elongated. So your parenchyma cells is the most common tissue. It is used in your packaging, major component of all the organs. It is very thin walled. ICS, interstellar space, is very less or completely absent. Various shapes, that is, it is isodiametric. But apart from isodiametric, it is spherical, oval, round, polygonal, or elongated in nature. It is elongated in nature. Now, this is where the question will come from. What are the functions? What are the different functions of your parenchyma? The first function is your storage as usual. The main function of your parenchyma is storage. Now, listen to me very carefully. Some of the parenchyma can become photosynthetic. That is, they can have chloroplast in them. So what do we call them? What do we call such cells? Such cells are called as your chlorenchyma. So they can be what? Photosynthetic. They can be photosynthetic. We call them chlorenchyma. We call them chlorenchyma. Now, some of the cells can become gas filled. They can become completely gas filled. Certain parenchyma can become full of gas. That is, they can become what? They take up gas and they help in floating. They help in floating of your particular type of your plants. That is called as your buoyancy. That is called as your buoyancy. Now, do certain type of your parenchyma can also help in gaseous exchange. They can also help in your gaseous exchange. Oh, aerenchyma, fold right. It's called as what? Aerenchyma. It's called as what? Aerenchyma. That is your help for buoyancy. Now, some of the cells can become what? Help in exchange of gases. Gas exchange. They help in gas exchange. That is called as your spongy parenchyma. They help in what? Spongy parenchyma. Spongy parenchyma. Now, one point is missing. The main point here. Is this a living cell or is it a dead cell? The answer is very simple. The parenchyma is a living cell. Parenchyma is a living cell. It is a living cell. Parenchyma is a living cell. I think one more point is missing. There were 5 points if I remember. Yes. The fifth point here. I'll write here. The fifth point here is the parenchyma can become secretor. They can secrete something. They help in secretion. That is, they can secrete some type of liquids. They can secrete some type of liquids. So what is a parenchyma? Parenchyma is a living type of tissue. Yes, it is a most common tissue. Most common tissue. It helps in packaging. Major component of all cell organelles. Very thin walled. Intercellular space are less or completely absent? What about the various shapes, that is spherical, oval, round, polygonal, or elongated in nature? Then we have functions is your storage, photosynthesis called as chlorenchyma, buoyancy by aerenchyma, gas exchange by spongy parenchyma. Also the secretions. Some of the parenchyma can become secretory in nature. So students, are all of these points clear? If you have any doubt about parenchyma, you can ask me right now. Any type of question about parenchyma, ask me right now. Madhumita is saying, yes, buoyancy is seen in the case of your aquatic plants, such as your pistia and eichhornia. They float, right? They float on the plant. They float on water because of the aerenchyma. They becomes gas-filled. Aerenchyma is gas-filled, so helps in buoyancy of the plants in aquatic, in water. Okay, example is your pistia and eichhornia. Any doubt here, students? Venti, Pandu, any doubt? Ask me right now. Any doubt will be cleared live. I do not wait for any of them, like, me going back, reverting. Any doubt, ask me live and I'll solve it in front of all of you. So if there is no doubt in parenchyma, can we do collenchyma? Can we do collenchyma? Yes, sir, we can do collenchyma now. So students, all of you to understand the first point here is your collenchyma is a living. It is also living. It is a living mechanical tissue. It is living mechanical tissue. Living mechanical tissue. Yes, it is a living mechanical tissue. So what is meant by isodiametric? Very good question. Iso means same. Iso means same. Diameter, a circle will have a diameter. Yes, this is a diameter. Entire thing is a diameter. The meaning of iso means same. This particular cell will have same radii, same diameter on all the sides. Same diameter. Here, this is a diameter, this diameter and this diameter is same in the cell. That is why they are called as isodiametric. That is they are called as isodiametric. Clear? All of you keep asking the doubts. I can always clear your doubts. Because students, if you watch it recorded, the doubts might not get cleared. You can always type in the doubts. Always type in the doubts. But I believe in, if a doubt should come in your head, you should solve it like that. Okay. Now, it is a living mechanical tissue. Now, what about intercellular space? Can you see anywhere intercellular space? No. The intercellular space is completely absent here. Yes. Intercellular space is completely absent. That is, there is no intercellular space. Intercellular space is completely absent. Now, what about cell wall here? What about the cell wall? The cell wall here is, the cell wall here has something called as your pectin depositions. It has something called as your pectin deposition. Pectin deposits. It has something called as your pectin deposits. Pectin is deposited in the cell wall here. Pectin is deposited in the cell wall. Pectin. T-I-N. Pectin depositions. Now, but where do you find them? Where do you find collenchyma? Collenchyma is usually found below the epidermis. It is usually found where? It is found below the epidermis. Found below epidermis. Below the epidermis is called as your hypodermis. Yes, below the epidermis is called as hypodermis. It is present in the hypodermis of your dicot stem. Of your dicot stem. Now, one question here is, this particular collenchyma, is it present in the case of the roots? Is it present in the case of your monocots? The answer is very simple. The collenchyma tissue is completely absent in the case of your roots as well as in the case of your monocots. That is the most important point here, that is the collenchyma. The collenchyma is completely absent. Completely absent in the case of your roots, absent in the case of your monocots. Absent in the case of your monocots. Collenchyma is absent in the case of roots. Collenchyma is completely absent in the case of your monocots. Monocots. The next point here, it is found in your, now, What about the vacuole? The vacuole in the case of your collenchyma is actually very large. Very large vacuole. The presence of vacuole is very large. They have a very large vacuole here. Very large vacuole. Very large vacuole. Now, do they have chloroplast? They are living cells. They have chloroplast. They are living cells and they have chloroplast. Living cells and they have chloroplast. So what is the function of your collenchyma? Very simple. Write down with me. The function is very simple. Let me erase this. Let me erase that and let me write it here. This is present in your hypodermis. Hypodermis in the case of your dicot stem. Now what is the function here? The function is, first one is very simple. I told you there is chloroplast. If there is presence of chloroplast, can the collenchyma tissue do photosynthesis? The answer is yes, sir. They can do photosynthesis. They can do what? They can do photosynthesis. They can do what? Photosynthesis. Now, what about the second point? I told you it is a living mechanical tissue. So, can it provide mechanical strength? Yes, sir. It can provide mechanical strength. Provide mechanical support or strength. Mechanical support. Now, third point is very important. The third point here is it provides elasticity. Provides elasticity to plant. So, what is the meaning of elasticity? Have you bent the petiole once? You know the petiole, right? Try bending the petiole. It will come back up. Try bending a small green stem. The green stem will become very elasticy, right? That is where collenchyma is present. So, where is collenchyma? Collenchyma is mainly present in the green stem as well as petiole, in the petiole of the leaf. What is petiole? Petiole is the stalk of the leaf. Stalk of the leaf is called as petiole. So, collenchyma is present in petiole, and in the case of your stem, it is completely absent in the case of roots as well as the monocots. There is no intercellular spaces. Cell wall is pectin deposits there. There are pectin depositions in cell wall found below the epidermis, that is in hypodermis of your dicot stem and leaf. Then we have large vacuoles and chloroplast is present. What are the functions? Function is nothing but your photosynthesis, mechanical support, and providing elasticity to the plant. That is your collenchyma. Any doubt here? Ask me right now. Any doubt about your collenchyma? I will clear it right now, here and now. Now, next is your sclerenchyma. Yes. Next is your sclerenchyma. Now, sclerenchyma is again a simple tissue. It is a simple tissue, but sclerenchyma is a dead tissue. Unlike your collenchyma, unlike your parenchyma, sclerenchyma is completely dead. It is completely dead. That is, can I say without protoplast? Without protoplast. Because protoplast is the living part of the cell. If there is no protoplast, the cell tissue is dead tissue. Now, what is the main function here? The main function is your mechanical support. Main function is your mechanical support. Main function is your mechanical support. Now, I told you in the case of your collenchyma, it was pectin depositions. Yes, pectin depositions were there in collenchyma. But in the case of your sclerenchyma, there is lignin depositions. What is there? Lignin deposition. Say it with me. Sir, the cells here are very long. The cells here are very long. Cells are narrow. Yes, sir. The cells are very long here. Cells are very narrow here with lignin deposition. Lignified. They're lignified cell walls. Lignified cell walls. Now, this sclerenchyma here can be further divided. Sclerenchyma can be further divided based on your function. Scleren can be further divided into based on the function, origin, and structure. Your sclerenchyma can be divided based on your function, origin and structure into two types. That is nothing but your what? Fibers and sclerates. Fibers and sclerates. What are fibers? Fibers are nothing but your thick-walled, elongated cells. Yes, they are very thick-walled. See, can you see? Thick-walled, elongated cells generally occur in group. They occur in multiple pairs, and their cell wall has pits. It has pits. It can be simple or bordered. Simple or bordered. Now I'll teach you what are pits, but give me a minute before I explain you what are sclerates. What are sclerates? Sclerates are very highly thickened cell wall. Sclerates are very high thick cell wall. Yes, very high thick cell wall. The shapes can vary. They have very narrow cavity or lumen. Here, this is your lumen. Lumen is said to be obliterated. Means lumen is completely filled here. Completely lignified. Lumen is completely lignified in the case of your sclerates. Now, this is a previous question. Listen to me very carefully here. Found in the case of your fruit walls of walnut, fruit pulp of guava and pear. Where are they found? Sclerates are found in the case of your fruit walls of your nut, fruit pulp of guava and pear, and in the seed coat of your legumes as well as tea leaves. There, in those places we can find sclerates. Now, what is the meaning of pits? Very simple. This is your cell wall. This is the plasma membrane. This is the cell wall. This is the plasma membrane. Now, between here we have the lignin deposits like this. Lignin deposits are there. Yes, sir. It's completely lignified. I told you it is completely lignified. But in between the lignification of the cell wall, there are small gaps. Here. In the lignin deposition, in your cell wall, there are small gaps. Those gaps are called as your pits. Gaps are called as what? Pits. Clear? We have your cell wall, plasma membrane. Inside we have your gaps or pits. That is the difference between fibers and sclerates. Sclerates are more lignified. Sclerates are more and more lignified. That is, they have very narrow lumen, and they are found in the case of your nut walls of your nuts, fruit pulp and guava and pear, and seed coat of your legumes and tea leaves. And so what is the role of pits? Pits are a small location where a breathing space becomes, like a small content can, like small secretions can exit out, small secretions can come inside. Yes, it becomes a narrow channel. Okay. Now, can we solve a question? All of you? Which of the following is not a character of your colon-chyma tissue? They provide mechanical support to the growing part. Yes, it is a property. They occur in layers below the epidermis. I told you, they occur below the epidermis in the hypodermis in dicot plants. Yes. They consist of cells with thick corners due to cellulose deposition. Yes. They have cellulose as well as pectin. They are usually dead and without protoplast. Is colon-chyma living cell or the colon-chyma dead? You tell me. Colon-chyma is living cell. So this is a incorrect statement. That is the incorrect statement right here. Okay. Next question. This is a NEET 2022 question. This is also a NEET 2022 question. The type of tissue commonly found in the fruit walls of the nuts. I'll give you 10 seconds to answer the question. Which is the tissue which is found in your fruit walls? 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. The answer is your sclerets. I told you just now. Sclerets are the structure which is found in the pulp of your fruit nuts or pear or tea leaves. Yes, here. Didn't we mention already here? See here. Found in your fruit walls of your nuts, fruit pulp or guava, pear, seed coat of your legumes and tea leaves. Or tea leaves. Clear? Is that point clear to all of you? Can we start with complex tissues now? Any doubt in the case of your simple permanent, ask me right now. I'll give you 1 minute or 2 minutes to ask all your doubts regarding your simple tissues. If there are no doubt, absolutely all of you, if all of you love the session, if you're liking the session, please like the video and also subscribe to the channel. Do not forget to subscribe to the channel. And we will start with your next topic, that is your complex tissues. Is everyone following with me? David, crazy? Yes. MED, Bhavani, Ravindran asking, can you please explain the function of? 1 second, I cannot see it. 1 second. Function of your pits. The pits have simple function. Wherever there is no cell wall, there are pits. Pits are basically small channels. Pits are basically small channels. What is someone asking me, yes sir, if they were to given sclerenchyma fibers, see, here, this is where so many students went wrong. Here they have given you, where is the question? They have given you sclerenchyma and sclerites here. Now, if this question, if sclerites was not given, if fibers was given here, then you should choose sclerenchyma. But now sclerites is given here. This is a more accurate answer. So this is correct here. This is correct now. Okay? Clear? All of you? David, duration of class is going to be a little long because I am teaching you in detail. This is the most detailed class. You are making the notes with me, right? You are making the notes with me, and notes will also be given to you later on. So I'm taking it slow so that all of you can understand and not by heart all this. Okay? Now, complex tissues. What is the meaning of complex tissues now? Can we start now? Can we start, students? I want green hearts in the chat, fire in the chat, hearts in the chat, anything possible in the chat, and I will teach you complex tissues. Now, I told you, complex tissue is also a type of your permanent tissue. Yes, permanent tissues stop dividing. So, can I tell your complex tissue also, they are not dividing. They are not dividing. Yes, sir, they are not dividing. They also have very specific function. They have very specific function. Very specific function they have. Very specific function. Then, what about the cells here? In the case of your simple tissue, all of them are same cell. But in the case of your complex tissue, we have different types of cells. We have different types of cells. We have different types of cells here in the case of your complex tissues. Students, in your NCERT, we have two main types of your complex tissues, that is your xylem as well as your phloem. Can we start? Hello Gopika ma'am. So can we start with xylem and phloem? So if you see a complex tissue, we have your xylem and we have phloem. Xylem can be a primary xylem, secondary xylem. Similarly, phloem can also be of primary as well as secondary in origin. Primary or secondary origin, that is phloem. This I'll tell you later. This proton meta I'll tell you later on. Now write down. Now write down with me, what are the different properties of your xylem? What are the different properties of xylem? Now xylem, all of you know, xylem is a type of your conducting tissue. Yes sir, xylem is a type of your conducting tissue. It is a type of your conducting tissue. Now, this xylem, is living or non-living? Simple question. Is the xylem living or non-living? It is actually both. Your xylem is both living and as well as non-living. Living as well as non-living. Some of the xylem elements are living. Some of the xylem elements are actually non-living. Some of the xylem elements are living and some of them are non-living. Now you tell me in the chat, xylem helps in conduction of water or food. Basic biology, basic botany, xylem helps in conducting water and minerals. It conducts water and minerals. Water as well as minerals. But where does the conduction happen from? The conduction actually happens from here, from the root to the shoot. From the root to the shoot, the conduction of water happens. Is this unidirectional or is it bidirectional? The answer is very simple. The xylem always conducts water in a unidirectional manner. That is from roots to the shoot. It is unidirectional. Unidirectional flow of water is happening here. Okay, unidirectional flow of water is happening here. Now we have different type of xylem, right? Different types of xylem. That is, you have tracheids, we have vessels, we have xylem parenchyma as well as xylem fiber. Tracheids, vessels, xylem parenchyma and xylem fibers. Now a simple question. I told you your xylem can be living as well as non-living. How many components here are living and how many components here are non-living? Tell me fast. Anyone can tell me in the chat how many of them are living and how many of them are non-living here? The non-living are here. Tracheids are non-living. Vessels are non-living. Parenchyma is living and your xylem fibers are non-living. So living to non-living ratio is 1:3. One of them is living, that is xylem parenchyma is the only living tissue here, but your vessels, tracheids and xylem fibers are completely non-living. They are completely non-living. So 1:3 ratio, 1 being the living, 3 being the non-living. Can we do about tracheids now? So that is just the basis of xylem. But let's understand in detail one by one now. One by one. If you look at the tracheids, the first one, they are the elongated tube-like cell. Yes, can you see this diagram? They're elongated tube-like cells with tapering end. This is called as what? Tapering end. Elongated cells with tapering end. This is called as what? Tapering end. Now, they have thick lignified walls. Meaning is what? The cell wall has a deposition. Which deposition? Lignin deposition is found in the case of your tracheids. That's why, because of the lignin deposition, these cells are completely non-living. Because your lignins will completely block the exchange of any material. Exchange of material is stopped. Right? That is why its lignin deposition is there. No chloroplast, nothing, dead cell. Then we have lignin deposition. They have inner layers of the cell walls have thickening that vary in form. So they have different type of thickening. For example, some of them can have ring thickening. Then some of them can have round thickening, ring and round. Dead cells without protoplast. Dead cells without the protoplast. One of the main water transporting elements of your xylem. One of the most water transporting element of your xylem. That is nothing but your tracheids. What are tracheids? Tracheids are elongated tube-like structure with tapering end, dead without protoplast. Inner layer cell walls have thickenings. They have lignified walls. They have thick lignified walls. That is the entire notes which will be given to you on the Telegram channel about tracheids. Everything about tracheids is right here. Now, what about vessels? Vessels are living or dead? The vessels are actually dead cells. They are also what? Dead cells. That is no protoplasm. There is no protoplasm means the cells are completely dead here. Now what about how are they made? Are they tube-like? Yes sir, they are tube-like here. That is long cylindrical tube. Can you see this tube here? Long cylindrical tube here, and they are made up of something called as many cells. They are made up of many cells here. Can you see this? This one of them here. This one of them is one cell. This is another cell. Oh, I forgot to mention. Hello, I forgot to mention these vessels are actually multicellular. They are what? Multicellular. And your tracheids are unicellular. Almost forgot to mention this. They are unicellular, while your vessels are multicellular. That is, they are made up of your vessel members. Each one is a vessel member, and one after another they are arranged like this. One after another they are arranged like this, and between them they have something called as perforated wall. Can you see this here? Each cell has lignified cell wall and central cavity. Inside the central they have what? Central hollow cavity through which water is passing, and each one is joined like this. Each one is joined like this, interconnected through the perforations in their common wall. Perforations means small holes. There are small holes present here. That's how they are connected throughout. They are multicellular in nature. One after another they are joined. One after another they are joined. Typical feature in the case of your angiosperms. It is seen mainly in the case of your angiosperms. Clear? It is mainly seen in the case of your angiosperm, while your tracheids are seen in the case of your gymnosperms. Gymnosperms, or it could be your pteridophytes. Or pteridophytes. Clear? That is your tracheids. This is your vessels. That is long cylinders. They are multicellular. One after another they are joined. They are made up of many cells. Yes, multicellular. One after they are joined. Each cell has lignified cell wall and large central cavity. No protoplasm. Interconnected through the perforations. They have perforations in their common wall. In the common wall they have perforations. That's how they are interconnected. A typical feature in the case of your angiosperms. Difference you see? Tracheids are present in all vesicle plants. They are present only in your angiosperms. Narrow lumen, wide lumen. Highly thickened, less thickened than the tracheids. Your tracheids do not have end plates. I think Crazy was mentioning this. This is why I was waiting. Because here I have mentioned here, they do not have end plate. They have perforated end plates. Here, these are your end plates. These are your end plates are present. One after another they are arranged, and in the end they have something called as end plate. Okay, they have what? Perforated end plates, perforated end veils. Only in the case of your vessels they have perforated end plates, and the end plates are absent in the case of your tracheids. Do not have end plates. Clear? Then we have your parenchyma. Yes, this is your living cell. All of you know living cell. They have thin walls. Again, same like your parenchyma. Parenchyma is thin wall. What is the function of parenchyma? Storage, right? Cell wall is made of cellulose. Function is what? Storage of food material in the form of starch or fat and other substances like your tannins. Living cell wall, thin like your parenchyma. Cell wall is made of cellulose. Function is storage and substances like your starch, fats as well as tannins, just like your normal parenchyma, xylem parenchyma. Then we have your xylem fibers. Just like your fibers, they have highly thickened walls. Yes, highly thickened walls. Obliterated center lumen. What is the meaning of obliterated? The meaning of obliterated is it is completely filled in the center. Completely filled with what? Lignin. Completely highly thicnified lignin is present there. They can be septate or aseptate. Septate meaning they have sectioning, they have cross sectioning. If it is septate, it has cross sectioning. Aseptate, no cross sections. Aseptate, no cross section. Clear? Ask me right now, any doubt till now? Any doubt till now, ask me right now. Your xylem can be of your primary xylem or secondary xylem. What is primary xylem? Primary xylem is a xylem which is present in the starting stage of your plant. That is protoxylem and metaxylem. Protoxylem is first formed primary xylem. It is first formed. While your metaxylem is later formed. Metaxylem is later formed. Okay. Now based on this information, I'll teach you one more concept. Based on this information here, I will teach you one more concept. That is nothing but your primary xylem can be divided. Primary xylem can be divided into your endarch and exarch. What is the meaning of endarch and what is the meaning of exarch? The meaning of your endarch is, can you see this? This is your pith. This is your pith. This is your pith. The meaning of endarch. The meaning of endarch is end. End means endo. Endo means inside. If your protoxylem, if the protoxylem is towards the pith, if protoxylem is towards the center or pith, it is called as endarch. It is called as endarch. Now what is exarch? Ex means outside. Exosphere. Exosphere, right? So it towards outside. If your protoxylem is towards the periphery or protoxylem is towards the X outside, it is called as your exarch. It is called as exarch. Now what is endarch? In endarch, endo and endarch, protoxylem is towards inside. Now what is exarch? X means outside. Like how some of you might have girlfriends and boyfriends, right? X is out of your life, right? X is out of your life. What is the meaning of that? Meaning of that is your protoxylem is towards the outside here. Protoxylem is towards the outside. Exarch towards the outside. Protoxylem towards the outside. Now exarch is seen in the case of your roots. Exarch is seen in the case of roots, and endarch condition is seen in the case of your stem. Endarch is seen in the case of your stem, and exarch is seen in the case of your roots. Will you remember this? Will you remember this? Tell me in the chat right now. Will all of you remember this? Endarch means your protoxylem is towards the center, endo inside. What is the meaning of X? X is exarch means outside. Your protoxylem is towards the outside. Clear? All of you will remember this? Amazing. Now, solve this question. Can you solve this question, all of you? This is a question from your NEET 2023 question. Solve this question. Let me sit for 10 seconds while you solve this question. What does the question say? Statement number 1. Endarch and exarch are the terms often used to describe the position of your secondary xylem in the plant body. Exarch condition is most common feature in the case of your roots. Exarch in the case of roots. Now you tell me in the chat. What is the answer? Statement 1 is correct and statement 2 is also, statement 1 is incorrect, but statement 2 is correct. Both statement 1 and statement 2 are true. Both statement 1 and statement 2 or both are false. Statement 1 is correct, but statement 2 is false. 10 seconds. Take your time. Answer. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. The answer is your first one. That is, statement 1 is incorrect. Why do I say so? Because students, listen very carefully. Endarch and exarch are the terms often used to describe the position of your primary xylem. Here, primary xylem gives endarch and exarch, not the secondary xylem. Secondary xylem comes in your secondary growth. Okay, this is your primary xylem which is dividing. Clear with all of you? Is everyone following the questions? Understanding the questions? Now, let's start with phloem now. Let's start with phloem. After the phloem, in 10 more minutes, I'll finish phloem. Then I'll give you a 10 minutes break. After phloem, 10 minutes break. All of you go back and then we will do your tissues, ground tissues. Okay. Now, start with me. Phloem. It is also a type of your conducting tissue. It is also a type of your conducting tissue. Yes, sir. It is a type of your conducting tissue. Now, students, you tell me in the chat. Does the phloem helps in conducting water or does it help in conducting the food? You tell me in chat right now. This particular phloem helps in conducting the food. Helps in conducting the food. Also, can I also write as helps in transport of food. Helps in transport of food. Now, where is the food material? Where is the food material synthesized? It is synthesized in the case of your leaves. That is light reaction and dark reaction. After dark reaction, we have the food. So your food is present in the leaf. Now from this leaf, the food has to go to your, imagine there is a flower on top. The food has to go to flower. The food also has to go to the roots. So can I call this type of arrangement or this type of flow as bidirectional flow? Yes, it is also called as your bidirectional. Bidirectional. It is bidirectional. It is going from your flower also to the shoot also or to the root also. It is bidirectional in nature. Bidirectional in nature. Now, phloem has again 4 types. That is your sieve tubes, companion cells, phloem parenchyma and phloem fibers. Now again, students, tell me in the chat right now, how many of the following are your living and how many of the following are non-living? Tell me first in the chat. Your sieve tubes are living, companion cells are living, parenchyma is living, and only, that is, phloem fiber is non-living here. Only the phloem fiber is non-living. Again, it is your, it's now 3:1 ratio. 3 is your living and 1 is your non-living. 1 is your non-living. Clear? Now let's study one by one. Let's understand sieve tubes and sieve elements. Your sieve tubes and companion cells together, because they are also called as your sister cells. They are called as what? Sister cells, because these cells are ontogenetically connected. Now what is the meaning of ontogenetically connected? It is said that both your companion cells as well as your sieve tubes arise from a same mother cell. So they are like your sister cells. They are like your sister cells. That is sieve tubes as well as companion cells. Now what are sieve tubes? They are long tube-like structure. Yes, they are long tube-like structure. Here, can you see this one? Here, long tube-like structure. They are long tube-like structure. This is your sieve tubes. They are arranged longitudinally. Yes, they are arranged longitudinally, associated with companion cells. So here it has a sister. Your sieve tube or sieve elements has a sister. The sister is your companion cells as the sister here. Okay. Now end walls are perforated in the sieve-like manner. The end walls here. The end wall. Can you see this? This is the end wall. The end walls are perforated. Can you see holes? Small holes here. Yes, sir. I can see small holes here. So the end walls here are perforated. Perforated end walls. Mature sieve elements have a peripheral cytoplasm and large vacuole and lacks the nucleus. They have what? Large vacuole for storage, for transportation, but they have lacks nucleus. So this is a very important point. In the case of your sieve tubes or sieve elements, the nucleus is completely absent. That is why functions is controlled by your nucleus of the companion cells. Just like your, when you do not have your food, when you don't have your pen to write, what do? You go to your sister's room, you go to your brother's room, you take their pen and come, right? You take their notebook and you start writing. That's what sieve tube is doing. Sieve tube does not have its own nucleus. It is using the nucleus of your companion cells. Functions is controlled by your nucleus of the companion cells. What are companion cells here? They are specialized parenchymal cells. Yes, they are closely associated with the sieve tube elements. The sieve tubes and companion cells are connected by the pit fields present between the common longitudinal wall. In this common longitudinal wall, there are pits here. There are pits here. That's how they are connected. There are small pits there. That's how they are connected. And what is the function? This is a NEET PYQ here. NEET PYQ right here. NEET PYQ, what is it? The maintenance of pressure gradient in sieve tubes. What is the function of your companion cells? It maintains the pressure gradient of the sieve tubes. Pressure gradient of your sieve tubes. Clear? Is that point clear to you? So what is pit fields? Pit fields are nothing but your channels. I told you, pits, lignified pits in the case of your sclerets. Sclerets also had pits, right? Pits, small pits, lignified pits. The same pits are present here also. Small pits are there. But in the small pits, what are pits? Pits are nothing but your small gap in the lignification. Small gap in the lignification is the pits. They have small pit fields here. That's how your companion cells and sieve elements are interconnected. Clear? Now your sieve elements are present in the case of your angiosperms. But in the case of gymnosperms, both of these are actually absent. In the case of your gymnosperms, they have something called as your sieve cells. In case of gymnosperms, they have something called as sieve cells in the case of gymnosperms. Also called as your, and it also has something called as albuminous cells. They have something called as your albuminous cells and as well as your sieve cells in the case of gymnosperms. But your companion cells and your sieve elements are only in angiosperms. Only in angiosperms. Clear? Albuminous cells are present in the case of your gymnosperms. Sieve cells are present in the case of gymnosperms. But in the case of your angiosperms, we have your companion cells as well as sieve tube elements. Okay? Sieve tube elements. Now, then we have your phloem parenchyma. Again, parenchyma is always same. That is elongated, tapering and cylindrical walls. See the diagram? Elongated, tapering in some cases here, tapering as well as cylindrical cells, dense cytoplasm and nucleus, large nucleus in parenchyma, absent in monocot. That is the most important here. What is the most important point here which is highlighted? Phloem parenchyma. Phloem parenchyma is completely absent in the case of your monocots. That can be a question. So it is absent in the case of monocots, that is phloem parenchyma. Plasmodesmal connections exist between the cells. Main function is again storage and other substances like your mucilage, resins and latex. Again, storage is the main function. Students, please note this. Absence in the case of monocots. That is your phloem parenchyma. Clear? Am I going too fast still? Am I going fast? Let me know if the pace is correct. Thumbs up in the chat. Okay. Then we have your phloem fibers. That is, they're made up of your sclerenchymal cells. It means they're living or dead. Obviously dead. Absent in your primary phloem. present in the secondary phloem. That is also very important line. Very important line, that is phloem fibers is absent in primary phloem. In the starting stages of the plant, we do not have your phloem fiber. But later on in your plant's life, phloem fiber will develop. Okay, it is elongated and unbranched. See the diagram? Pointed needle-like apices. Cell wall is very thick. At maturity, it loses the protoplast and becomes the dead cell. It becomes the dead cell of your phloem. The only dead cell of your phloem is your phloem fibers. What is the most important point here is present in primary, absent in primary phloem, present in your secondary phloem. It is made up of your sclerenchyma. Cell wall is very thick. It is loose protoplast and becomes the dead. It becomes completely dead here. That is the phloem parenchyma as well as your phloem fibers. Clear? Amazing, all of you are. Now we learnt about your phloem, primary as well as secondary. Primary is made up of your protophlloem, metaphlloem. What is protophlloem? First formed primary phloem is your protophlloem. Metaphlloem is later formed primary phloem is your metaphlloem. Now let's solve a question. Let's solve a question. Let's see if you're able to solve the question. Phloem gymnosperm lacks. What does it lack? This is your NEET 2019 question. NEET 2019 question. In the case of your gymnosperms, what does it lack completely? Is it your both sieve tubes and companion cells, albuminous cells and sieve cells, only sieve tubes, only companion cells? 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. It both in the case of your gymnosperms lacks both sieve tubes as well as companion cells. Lacks both of them. Instead of your companion cells as well as your sieve cells, it has something called as albuminous cells as well as sieve cells. Clear, all of you? All of you are able to solve the question? All of you are able to solve the question? Amazing. All of you are able to solve the question. The next topic here we are doing is your tissue systems. Next concept entirely we are doing is tissue system. We are completely done with your simple tissue, complex tissue, done and dusted. If you have any doubt till now, ask me. Students, sometimes if you are learning this chapter for the first time, if you are understanding this chapter for the first time, some of the concepts can become very overwhelming. But what you need to do here is, after I finish the class, please read the NCERT once. After that, please go through my notes. Please go through my notes because I cover everything in my notes. NCERT snips, NCERT lines, as well as previous year questions. So please download my notes and you should take advantage of it. Okay, please take advantage of my notes. Now, in your tissue system, we have something called as your 3 parts. Tissue system is mainly divided into 3 parts. The first one is your epidermal. Then we have your ground tissue. Then we have your vascular bundles or vascular tissue. So your entire tissue system is mainly divided into 3 parts. That is your epidermal, ground tissue, as well as your vascular tissue. 3 different parts of your tissue system. Now let's learn one by one. Let's understand each one of them. Can we start? Can we start, all of you? Now, see, this is your epidermal tissue system. In your epidermal, mainly has 3 different parts. Epidermal has 3 different parts. What are they? The first one is your epidermal appendage. Yes, on the epidermis of your shoot, on the epidermis of your root, we have something called as your epidermal hair. That is something called as your epidermal appendage. Then we have your epidermal cells, that is your outer covering cells. Then we have your stomata. Then we have your stomata. These three are the different parts of your epidermal system. Oh, there was a break, right? I totally forgot. So can we take a 10 minutes break, all of you? Do you want a 10 minutes break? 10 minutes. Now what is the time now? The time is 8:05. Let's start at 8:15. Let's start at 8:15, 10 minutes break. I forgot about the break. See, when you're teaching, right, even though you are somewhere, even though I don't even have a single student sitting in front of me, I just have a camera in front of me and the board here. And I completely forgot about the break. I completely forgot about the break here. Okay, so we'll take a break now. This is what happens when you're teaching, when you get in the whole zone. I just have a camera in front of me, nothing else. Okay, so time is now 8:05. Let's take a break till 8:15. Break till 8:15. Okay? Okay, all of you? So see you back in 10 minutes. Drink some water, go to the washroom, get something to eat also. Get something to eat. Okay? Get something to eat. I'll also take a 10 minutes break. I'll drink some water and come, and we will start after 10 minutes. Promise me you'll come back. Please come back, okay? This is what happens. You don't come back only. So welcome back each and every one of you. So let me know in the chat. Someone is asking me, sir, where are you from? I am from Bangalore. Let me know in the chat where you are from. So students, let me know in the chat where you're from. I feel, where is Shivani from? Where is Akansha? Where is Pandu from? Where is Ravindrani from? Everyone. So students, did you eat your dinner? Tell me first, did you finish your dinner? Because I did not, because I'll eat later. Did all of you eat your dinner? Amazing. Yes, we are back. Yes, we are starting with your epidermal tissue system. Now students, for the next 1.5 hour or 1 hour, next 1 hour will be your class. After that, trust me, you will understand. You will have the feeling that yes, I have made sure that I got the 14 marks or 16 marks. Okay. Telangana, yes, I know Ravindran is from Telangana, I remember. I am from AP, sir. Pandu is from AP. So don't worry, next 1 hour, we will finish the entire of your sectioning. So entire of your sectioning, I'll be teaching with the help of diagram. I won't be, obviously I'll show you diagram, I'll be writing it, then I'll be reading your NCERT lines. Okay. So before that, let's start with your epidermal tissue system. AP, okay, we have a lot of students from AP, that's amazing. Now, let's understand epidermal tissue system now. I told you your entire of your ground tissue system. Your entire of your ground tissue system has been divided into your epidermis, cortex and vascular bundles. Yes, epidermis, cortex as well as your vascular tissue. Now in your epidermis we have your epidermal appendages. Yes, we have the epidermal appendages. Then we have your epidermal cells. Then we also have your, let me erase this. Then we also have your, what? Also have your stomata. We also have the stomata. Now if you look at the epidermal appendages, what is the meaning of appendage? Appendage means an extension. Appendage means a extension which is present in the case of your root or shoot. Now in your root appendage, we have something called as your root hair. So can I also call it as a, this is a root hair is a root appendage. It is a root appendage. It is a root appendage. Now root hair is unicellular or is it multicellular? Your unicellular. Your root hair is usually unicellular. Root hair is what? Root hair is unicellular. Now students, tell me, root hair develops from which region? And in your morphology, I told you, in the case of morphology, I mentioned root hair develops from your region of your maturation. In the region of your maturation, that is also your epidermis. From the epidermal region of your region of maturation, root hair is developing. Now students, tell me, is this root hair exogenous or is it endogenous in origin? All of us know your root hair is exogenous in origin. It is exogenous in origin. It is unicellular, it is exogenous origin, it is a root appendage, that is root hair. Now what about trichome? Right, trichome is a type of your stem hair. Trichome is a type of your stem hair. Now again, is this produced exogenously or is it produced endogenously? Remember always, trichome is also produced exogenously. Trichome is always and always produced exogenously. Then, is it multicellular or unicellular? Your trichomes can be both actually. Majoritarily they are multicellular, but sometimes they can be unicellular. Majoritarily they are multicellular, but sometimes, by majority are multicellular, but sometimes they can be what? They can be unicellular. Sometimes they can be unicellular in nature. Now, what are other properties here? What is the function of your trichome? Trichome actually controls the water loss. Trichome helps in water loss. It is controls water loss. It controls water loss. Water loss by your transpiration. Transpiration. It reduces the water loss by transpiration. Then we have the other point here is this trichome can be soft or hard. Trichome can be soft or hard. The same trichome can have certain lines. The trichomes can have certain lines like this. Have you seen trichomes anywhere? Tomato plant. How many of you seen tomato plant? On the tomato plant, try touching the leaf. If you touch the tomato plant, now touch the leaf of it, you'll have the small hair kind of on tomato plant, on the leaf, on the stem, tomato plant. That is your trichome. That is the trichome. It can be septate or aseptate. Septate or aseptate. Now this trichome sometimes can become what? This trichome can sometimes becomes very secretory. It can produce certain substances. So can I also say, but this is my spelling, my handwriting, they can become secretory. They can become what? Secretory. Become secretory in nature. They can secrete few small substances. That is your trichomes. So we have your epidermal hair, we have your root hair, then we have trichomes. Root hair, we have root appendages, unicellular, exogenous. Then we have your trichomes, which can be branched. Can you see? They can be branched or unbranched. Stem hair, exogenous, multicellular, but sometimes unicellular. Control water loss of the transpiration. They can be soft or hard. They can be septate or aseptate. And finally, they can also be secretory in nature. Secretory means what? They can release certain substances. That is your nothing but your epidermal hair. Now let's understand what is epidermis. I will make you write down certain points which are going to be most important for your making your own notes. Okay, let's understand different points about your epidermis. Now tell me, epidermis is present on the outside or inside? Epidermis is the outside layer. Yes, all of you agree with me? Epidermis is the outermost layer. Yes, sir. Epidermis is the outermost layer in a tissue. Yes, epidermis. Now, in the epidermis, do you see any intercellular spaces here? No, sir. I cannot see any intercellular space here. So, can I tell, in the case of your epidermis, intercellular space is completely absent. Right on with me. Intercellular space is completely absent here. Now, epidermis. Epidermis, is it a single layer or is it multiple layer? Remember students, here this was the epidermis. This is the first part. This is the epidermis. It is always a single layer. Epidermis is always a single layer. It is always a single layer. Now, what is the shape of the cells here? The shape of the cells here are barrel-shaped cells. They are what? Barrel-shaped cells. Can you see this? This is called as barrel-shaped. They are what? Barrel-shaped cells. Barrel-shaped cells. They are barrel-shaped cells. Now, what are they made up of? Remember this very important point. I told you parenchyma cells. Parenchyma cells are usually photosynthetic in nature, but in the case of epidermis, or epidermal cells, epidermis is made up of your parenchyma. But can it do photosynthesis? The answer is no. This is a tricky part here. This particular epidermal region or epidermal cells are made up of your parenchyma. They are made up of your parenchyma cells. They are made up of parenchyma cells, but they do not do photosynthesis. They do not do what? Photosynthesis. Can I write chloroplast is absent here? Chloroplast is completely absent here. Chloroplast is completely absent here. So no photosynthesis. Photosynthesis is absent here in the case of your epidermal cells. Except, this is very important now, except your guard cells. Except the guard cells. So on the epidermal cells, there are certain regions where we have green coloration. In epidermal cells, there are small regions we have some green color structure called as your guard cells. They do, yes, Madhumita, they do not contain your chloroplast. Chloroplast is absent except the guard cells. Except the guard cells. In the case of your epidermal cells, the chloroplast is completely absent. Chloroplast is completely absent. Intercellular space is absent. It is a single layer made up of parenchyma, barrel-shaped cells, and they are the outermost layer. They are the outermost layer. Now, what about the vacuole? Do they have vacuoles here? Yes, sir. In the case of epidermis also, sometimes we can see very large vacuoles. Sometimes we can see what? Large vacuoles. Large vacuoles. We can see large vacuoles. Now, one more point is missing here. The one more point which is missing here is on top of your epidermis, on top of the epidermis of your leaf and stem, we have a waxy coating on top of your epidermal cells, which is a barrel-shaped cell. We have a waxy coating. We have a waxy coating called as cuticle. Waxy coating called as cuticle is present. We have a waxy coating on top of your epidermal cells which can prevent water loss. That is called as your cuticle. Amazing, all of you learning? Point is clear. Epidermal cells are barrel-shaped cells made up of parenchyma, single layer, no intercellular space, outermost layer, chloroenchyma is absent except your guard cells, large vacuole. On the top of your epidermal cells, we have waxy coating, which is called as your cuticle. Amazing. All of you are amazing. We learned just now. Appendages is over. Epidermal cells is over. Finally, we come to the stomata. Now, I told you on certain regions of your guard cells, on certain regions of your epidermal cells, we have something called as your guard cells. Now, if I want to draw that guard cells, this is your stomatal aperture. In the case of dicots, we have something called as a bean-shaped. In the case of your dicots, we have bean-shaped guard cells. This is your guard cells. This is your nothing but your core or aperture. Or small hole. Aperture. Now, surrounding the guard cells, we have certain epidermal cells. Surrounding the guard cells, we have certain epidermal cells. Now, these epidermal cells are called as your subsidiary cells. This is your epidermal cells, which are called as your subsidiary cells. They are called as what? Subsidiary cells. They are also called as what? Subsidiary cells. So we have your stomatal pore or your stomatal aperture or core. We have bean-shaped or kidney-shaped guard cells. Surrounding the guard cells, we have your epidermal cells, which are called as your subsidiary cell. Now, this particular, I told here, this waxy coating is present in the case of your leaf and stem. It is absent in the case of your roots. It is absent in the case of your roots. Now, guard cells, subsidiary cells, done. Now, this is a question here. In case of your monocots, it is dumbbell shaped. The guard cells are dumbbell shaped in the case of your monocots. But in case of dicots, it is bean shaped. We have your stomatal aperture, then we have the guard cells, then we have subsidiary cells. Together, all three of them guard cells plus stomatal aperture plus subsidiary cells, they are called as your stomatal apparatus. They are called as what? Stomatal apparatus. What is stomatal apparatus? Subsidiary cell plus stomatal pore plus the guard cells. In total, they are called as stomatal apparatus. Okay. Now, the ground tissue. Epidermis is over. Now, let's understand the ground tissue. Now, ground tissue has many parts. For example, all tissues except epidermis and vascular bundle. What does come under ground tissue? Except the epidermis and vascular bundles, everything in the middle. Everything in the middle is part of your ground tissue system. They're part of your ground tissue system. For example, the first one is your cortex. The first one is your cortex. Then we have your pericycle. Then we have your pericycle. Yes. Then we have pith. Then we have pith. Then we also have medullary race. Then we also have something called as medullary race. All of these are parts of your ground tissue system. Ground tissue system. Now in the cortex, below the epidermis, we have the hypodermis. Below the epidermis, we have the hypodermis. Hypodermis in the case of your stem. In the case of stem, we have the hypodermis. Below the hypodermis, in the case of your cortex, we have the general cortex. General cortex, also called as your main cortex. Also called as what? Main cortex. Then we have your endodermis. Endodermis. Now students, please remember, endodermis is part of your cortex. Endodermis is part of your cortex. Once again, someone is asking a question. Let me see the question. If you type big question, right, the big questions are not visible usually. Where is the mouse? Madhyamita sir, then among all the tissue system, ground tissue is the majority. Yes, ground tissue is the majority of them. So ground tissue has your cortex, pericycle, pith and medullary race. Cortex is divided into your hypodermis. Below the epidermis is the hypodermis. Below the hypodermis we have the general cortex or main cortex. Below the main cortex we have the endodermis. Then we have your pericycle, where from the pericycle what happens? From the pericycle lateral roots arise. Lateral roots arise from the pericycle. Then we have the pith. Then we have the medullary race. These are the major constituents of your ground tissue system. See here, ground tissue system in the case of stem and root is what? Made up of your parenchyma cells. In the case of leaves, it is mesophyll cells, that is thin-walled chloroplast containing cells. Again, mesophyll cells are also a type of your parenchyma cells. See here, cortex, pericycle, medullary race and pith. In your cortex, what do we have? In your cortex, we have your normal, that is your hypodermis, ground main cortex, as well as your the lowermost one, that is nothing but your endodermis. So please remember students, endodermis is part of your cortex. Endodermis is part of your cortex. Once again, please repeat after me. Some of you always forget this. I've been telling this from almost so many years. Still students ask me, sir, is endodermis part of your cortex? Yes. Please say after me, sir, endodermis is part of cortex. Ray, endodermis is part of cortex. Say after me that one at least. Never forget that. Never forget all of that. Okay. Now, let's start the last part of your tissue system, that is your vascular tissue system. Can we start? If you have no doubt till now, please quickly show me some emojis, then I will know you do not have any doubt till now. Can we start? Can we start with your vascular system? So, vascular system is very simple. Vascular tissue system is basically nothing but arrangement of your xylem and phloem. Basically, your vascular tissue system is nothing but your arrangement of your xylem and phloem. Arrangement of xylem and phloem. So your vascular tissue system is mainly divided into two types. One is radial, one is called as conjoint. One is radial, one is conjoint. Now what is radial? If this is the cell, inside the tissue, inside tissue, someone is asking me, sir, parenchyma cells are very thin, but in the case of pericycle, they are very thick. Because in the case of pericycle, it has more protection. It has a protection of your, protection. It protects your checkpoint, access checkpoint. It is making sure everything stays in compact. That is why it is since remember, every single time if there is small variations, right, that could be based on your adaptations or based on the particular function. So pericycle, in the case of your parenchyma, in the case of your pericycle, the function is different there. Based on the function, parenchyma can change. For example, in arenchyma is again changing. It is becoming very sponge air. In case of your chlorenchyma, it is becoming photosynthetic. In the case of pericycle, based on the function of protection, it is becoming thick-walled. Okay. The first one is your radial. What happens in radial? In the case of your radial, xylem and phloem. In the case of your radial, phloem, xylem. In the case of your radial arrangement of vascular bundles, the xylem and phloem are in two separate radii. This is the one radii. This is the separate radii. One second, let me show you. In the case of your radial arrangement, your xylem and phloem are present in different radii. They are present in different radii here. So can you see this? And they are different radii. Xylem and phloem are in different radii. And can you see they are in alternating fashion? Yes, sir. They are in alternating manner. They are alternating in manner. So can you see one phloem, one xylem, xylem. They are in different radii, and this is called as your radial arrangement of your vascular bundles, and this is seen in the case of your roots. Now let's understand conjoint. In the case of your conjoint, listen to me very carefully. In the case of your conjoint, xylem and phloem are present in the same radii. For example, we have, what do we have here? Xylem and phloem. Can you see? They are present in the same radii. That is nothing but your conjoint. This is seen in the case of your leaf and stem. Conjoint is seen in the case of your leaf as well as stem. Now, this particular conjoint is again further divided into two types. For example, if one is called as your collateral, the other one is called as bicollateral. Bicollateral is not in your syllabus, but I'm teaching you for you to understand. One is called as collateral, other one is called as bicollateral. Listen to me very carefully. In your collateral, what happens? In your collateral, we'll draw the same structure here. In the case of your collateral, xylem is present towards the center. See me, listen to this. Xylem is present towards the center. This is xylem, phloem, and phloem. This is your center. Xylem is present towards the center. That is called as collateral. That is called as what? Collateral. Now, sir, what is bicollateral? Listen to me very carefully. In the case of your bicollateral, xylem, phloem, xylem happens. In the case of your Sorry, phloem, xylem, phloem. Phloem, xylem, phloem. In the case of your bicollateral, what happens? Listen to me very carefully. In the case of your bicollateral, there is a sandwich. In the case of your bicollateral, there is a sandwich. Your xylem is sandwiched between two phloem. Phloem, xylem, phloem. Phloem, xylem, phloem. Phloem, xylem and phloem. That is called as bicollateral. There is a sandwiching between your xylem and phloem. Clear? Now, student, listen to me very carefully now. Your collateral is again divided into two types. Collateral is again divided into two types. One is called as your open. One is called as open. Other one is called as closed. Other one is called as closed. Imagine this is your vascular bundle. We have xylem here and phloem here. We have xylem here and we have phloem here. Right? Xylem is towards the center. Phloem is towards the outside. Now listen to me very carefully. In some cases, in the case of your stem, between the xylem and phloem, there is something called as vascular cambium is present. In the case of your stem, only in the case of monocots, vascular cambium is present between your xylem and phloem. Such type of vascular bundle is called as what? Open vascular bundle. It's called as what? Open vascular bundle. Now what if there is, between the xylem and phloem, if there is no cambium, it is called as closed. It is called as what? Closed. This is seen in the case of your leaf as well as monocots. Also monocots. Also in the case of your monocots. So let me quickly revise the entire thing. In your vascular system, tissue system basically tells you the arrangement of your vascular bundles. The first one is your radial. That is, xylem and phloem are in the different radii. See, this is one radii of xylem. This is the radii of your phloem, and they are in a alternating manner. That is present only in the case of your roots. Then we have your conjoint, where xylem and phloem are present in the same radii. Example is your leaf and stem. Now, conjoint have two types, collateral and bicollateral. What is collateral? In collateral, xylem is present towards the pith or the center. But in the case of your bicollateral, it is a sandwich. It is a sandwich between your phloem, xylem, phloem. Phloem, xylem, phloem. Phloem, xylem, phloem. Now your collateral is again divided into two types. If there is cambium, if there is presence of cambium, that is vascular cambium between the xylem and phloem, that is called as your open. That is called as your open. That is called as open. If there is no cambium present between xylem and phloem, it is called as closed. Closed is seen in the case of your leaf and monocot. If there is, this is seen in the case of your dicot. This is seen in the case of your dicots. And students, remember, this vascular cambium will later divide. This vascular cambium will later divide. That is why it is called as vascular cambium later provides your secondary growth. That is why this is seen in the case of your dicots. If there is no cambium, no secondary growth. Do you see secondary growth in the case of your leaf? No, sir. Do you see secondary growth in the case of monocots? No, sir. That is the answer. That's why in the case of monocots, there is no secondary growth, because they do not have the cambium between xylem and phloem. Clear, all of you? Is the point clear to each and every one of you? This is the entire vascular system. Please start the board series. Yes, we will be starting the board series. Don't worry. We are there for everyone. Lilac is asking me, open in the case of dicots, sir. Yes, open in the case of your dicots, closed in the case of your leaf and monocot. Flu. It is, see, how come in monocots? In monocots, it's always closed. Monocots, it is closed, because there is no secondary growth in case of monocots. That's why it's closed as closed. In the case of leaf and monocots, vascular bundles are closed. In the case of dicots, it is open. Is the point clear to all of you? Is the point clear to each and every one of you students? Clear? Pandu clear? Amazing. Amazing. Now, can we start? Can we, one question is there? What is the question here? Initiation of your lateral roots and vascular cambium during the secondary growth takes place in the cells of your pericycle. Happens in the case of your pericycle. Lateral roots and vascular cambium duration in secondary growth is pericycle. Direct question. Please, can you please explain again? Definitely. Why not? Quickly tell me. See here. You know what is collateral? In collateral, we have xylem and phloem in the same radii. Yes, xylem and phloem in the same radii. In the same radii. That is called as collateral. Now, students, listen to me very carefully. If there is a vascular cambium present between the xylem and phloem, if there is a cambium here, that is called as open. That is present only in the case of your dicots. If between the xylem and phloem there is no cambium, that is called as closed. No cambium, it is called as closed. That is seen in the case of your leaf as well as monocot. That is the meaning of open means cambium is present, closed means cambium is absent. Where is it absent? In the case of leaf and monocot. Clear all of you? Is the point clear? If you are still doubt, ask me. I am here for all of you. This is the channel where we do everything for all students. Next question. Oh, I didn't mention this, is it? Okay. Try solving this one. I didn't mention this point here, but I have added a question. I'll tell you what it is. Large, empty, colorless cells of the adaxial epidermis along the veins of the grass leaves is called as. On the leaves, we have large, empty, colorless cells. What are they called? They are called as your bulliform cells. I will be teaching you this right now. If you don't know, don't worry. I will be teaching you what is bulliform cells with the help of example. Let me take one piece of paper. Imagine if this is your leaf. Imagine, students, Nitya, Bhavani, if this is a monocot leaf. In the case of monocot leaf, what happens, right? If whenever there is less water, the leaf will turn like this. Leaf will become like this. Not like this. It will never become like this. The leaf will become like this. Can you see? The leaf will become like this to reduce the transpiration. That is nothing but your bulliform cells. Okay? That is nothing but your bulliform cells. The leaf will try to curve. Okay? That is because of your bulliform cells. Now, can we start with anatomy? Students, I know it has been taken a lot of time, but trust me, this is the most important part of this chapter. Now onwards, this is the most important part. I want each and every one of you to give you 100% now. We will be starting with your anatomy of your dicot as well as monocot. If I ask you, will a question come from here in NEET 2024? Yes. One question will definitely come. One question 100% will come from here. A sure short question. Everyone, I want every one of you to please share the session right now. Right now, share the session to all your friends. Ask them to attend at least this part. This part is going to be the most important part of the chapter. Okay? This is the most important part of the chapter. Please ask all your friends to join right now. Please like the video right now. And we will start with the most important part of the chapter. That is anatomy of your dicot as well as the anatomy of your monocot right now. Most important part. Okay? Can we start? The most important part of this chapter. First, we'll start with your students. I have diagrams. I'll teach you with the help of diagram. I think I have taught this in a similar fashion with the help of diagrams in my NCERT line by line. But now also, I will be writing each and every point about each and every layer. So I want each and every one of you to share the session right now, like the session, and I will teach you the most important part of the chapter. So we will be starting with dicot root. we'll be starting off with your dicot root. Now, the first point here, please repeat after me, write with me now. The first part of your dicot root is your epidermis here. The first part of your dicot root is your epidermis. Let's write a few points about your epidermis now. Students, I will be sharing these notes. I will be sharing these notes to each and every one of you. Please take advantage of this. The first part of your dicot root is your epidermis. Now students, this epidermis is a single layer. I told you already, epidermis is always single layer. See here, a single layer of epidermis. Now this epidermis is also called as, in the case of roots, epidermis is also called as epiblemma. It is also called as what? Epiblemma. What is it called as what? Epidermis is called as epiblemma. I know this can be confusing very fast. This can be confusing to so many students. So please focus now. There is epiblemma. Now students, epidermis is made up of parenchyma, collenchyma, sclerenchyma. I told you initially, epidermis is made up of your parenchymal cells. It is made up of what? Parenchyma. It is made up of your parenchyma cells. It is made up of your parenchyma cells. Now, if you notice the diagram, do you see any epidermal appendage? Yes, sir. I see epidermal appendage. That is nothing but your root hair. So there is presence of your root hair on the epidermis. So root hair is present here. Root hair is present here. Now I told you one point before. In the case of epidermis, in the case of roots, is the presence of cuticle. Do you have presence of cuticle here? No, sir. In the case of your epidermis of your dicot root, cuticle is absent. Cuticle is absent here. Cuticle is completely absent. Why cuticle is absent here? If there is cuticle, can the roots absorb water? Cuticle will block everything, right? If the cuticle layer is there, it will completely block. So there will be no absorption. Yes, that is why in the case of roots, cuticle is completely absent here. Cuticle is completely absent here. Now you tell me in the chat. Students, does the root have stomata? The answer is obviously no, sir. There are no stomata here. So can I write stomata are absent here? Stomata are what? Absent here completely. Stomata are completely absent. The last point here, which I already mentioned, in the case of epidermis, there is no intercellular spaces here. There is no intercellular spaces. Intercellular spaces are completely absent here. These are the following points I want every one of you to understand today. Epidermis, in the case of your dicot root, is single layer. It's called as epiblemma. It is made up of parenchyma. It has root hair on top of it. Cuticle is absent here. Stomata is absent here. Intercellular spaces completely absent in the epidermis of your dicot root. First part of your diagram completely decoded. Done. The next part is your cortex. The next part is your cortex. Can you see the cortex here? Now students, tell me, cortex here is green color. Cortex here is green color. Can I tell cortex is made up of your parenchyma here? Yes, cortex is made up of your parenchyma. Cortex is made up of your parenchyma. Now, do you see one layer of parenchyma or do you see many layers of parenchyma? I see many layers here. So there are many layers of parenchyma. So can I tell many or multi-layer? Many or multi-layer of parenchyma. Multi-layer parenchyma or multi-layer cortex is here. Multi-layer cortex. Clear? Now, do you see intercellular space here? Do you see any intercellular space here? In case of your parenchyma cells, there is little interstellar space now. You remember parenchyma, small interstellar space here. So all of you should write down here in your book your ICS is present, but it is very less. Interstellar space is present, but it is very less space here. Interstellar space is very less here. That is your cortex. Done. Next we go to your endodermis. The next part here is endodermis. So cortex is done, epidermis is done. Next is your endodermis. Write down here cortex. Then we have your endodermis. Now students, please remember the following points about endodermis. The first point your endodermis is actually part of your cortex, which I've been telling you from that time. What is endodermis? Endodermis is part of cortex. It is a part of cortex. Endodermis is part of your cortex. Now again, is it single layer or is it multi-layer? As MD is already saying, this your particular endodermis is single layer. It is what? Single layer. It is always single layer here. Single here. Now endodermis is made up of parenchyma, collenchyma, sclerenchyma. Endodermis is made up of your parenchyma. Just like your cortex, endodermis is made up of parenchyma. Endodermis is made up of what? Parenchyma here. Right? Now, the next point here I want all of you to understand here is this particular endodermis. This particular endodermis acts as a checkpoint. What is the checkpoint here, sir? When the water is entering through the roots to your shoot. When the water is entering, sometimes a foreign bacteria or a foreign particle can enter the soil. And from the soil, it can enter the roots. Now this endodermis acts as a checkpoint to make sure nothing extra is entering into the roots. So this endodermis acts as a checkpoint. It acts as a checkpoint. Acts as a checkpoint. Now in your endodermis, do you find intercellular spaces here? Can you see? In the endodermis, I do not see anywhere no intercellular space here. In the case of endodermis, intercellular space is completely absent here. Now, how is the shape of the cells? The shape of the cells here is again barrel-shaped cells. It is barrel-shaped cells. Barrel-shaped cells. They are barrel-shaped cells like this. Barrel-shaped cells. Now, within this barrel-shaped cells, on this barrel-shaped cells of your endodermis, there is certain type of depositions. These depositions are called as your subarene depositions on your lateral wall or as well as your tangential wall. This is a lateral wall as well as the tangential wall. There is depositions of your subarene. Depositions of what? Subarene. On the barrel-shaped cells, we have subarene deposition, which makes the Casparian strips. They have what? Subarene deposition. Now these subarene depositions will make up the Casparian strips. These subarene depositions will make what? Casparian strips. Where do I write it? I'll write here only. Casparian strips. Casparian strip. Casparian strips. Clear? They make the Casparian strips. Now students, if this is your barrel-shaped cells, the subarene will not be completely around it. Only in the this is the radial wall. If this is your barrel-shaped cells, only on the radial wall as well as the tangential wall. On the tangential wall as well as the radial wall, subarene depositions will be there. Not completely it will be there. Okay, not completely. So what are the properties of your endodermis? Endodermis is part of the cortex. Parenchyma act as a checkpoint, single layer, barrel-shaped cells, it has suberin deposition, which makes the Casparian strips. There is no intercellular space at all here. Cortex, parenchyma, multilayer and intercellular space is present here. Clear? Tell me every point here is clear as clear. Then we'll go to the next one. Listen to me very carefully. The next one is your, what is there next? After endodermis is done, cortex is done, epidermis is done. Then we have the pericycle. Then we have the pericycle. Then we have the pericycle. Now students, listen to me very carefully. Can you see pericycle is 1 layer, 2 layer, 3 layer? Can you see 3 different layers? So pericycle is actually a few layers thick. Pericycle is few layers. So it is made up of few layers. Pericycle is made up of few layers. Pericycle is made up of your few layers. Now again, pericycle is also made up of your parenchyma. It is also made up of your parenchyma. Parenchyma. See, this is your pericycle, few layers thick, but it is made up of your parenchyma. Now, pericycle has two main functions. I told you already. Pericycle can produce lateral roots. Pericycle can produce what? Pericycle can produce your lateral roots. In the case of your dicot root, lateral roots are produced by your lateral roots. Lateral roots are produced by your dicot root in the case of your pericycle. From the pericycle, lateral roots arise. And the second part, and remember, these lateral roots are endogenous in origin. They are endogenous in origin. Endogenous in origin. The second main function of your pericycle here is, that is, they are responsible for the secondary growth. Responsible for the secondary growth. That is, in the case of your pericycle, 1/3 of vascular cambium is there. No, this point is not important. Just remember this. That is extra information. I don't want to give you so much extra information. Remember this. For your NEET examination, this is what you should know. Pericycle is few layers thick, made up of parenchyma. Two main functions. It produces lateral roots. That is endogenous in origin. And it is secondary growth. It produces secondary growth. Now, do they do photosynthesis? The answer is no. No photosynthesis. Photosynthesis completely absent here. That is, chloroplast is absent here. Clear? Clear? Sir, parenchyma in epidermis of the root is photosynthetic. See, parenchyma in epidermis is not photosynthetic. BKN, the parenchyma in epidermis is not photosynthetic at all. Only in the guard cells, whenever there is guard cells, that region is photosynthetic. That's all. Apart from that, parenchyma in the case of your epidermis is not photosynthetic at all. Okay? No photosynthesis. Amazing, all of you are. Next one. Pericycle done. Then what is there next? Next is your what? Vascular bundles. The next one is your vascular bundles. Now vascular bundles are how? I told you, in the case of roots, in the case of root, do we have radial or do we have conjoint? I told you starting only. What is the meaning of radial? Xylem and phloem are in different radii. See here. This is your, where is it? Where is the phloem? This is your phloem. This is your xylem. Xylem and phloem. So are they in different radii? Yes, sir. They are in different radii here. So that is radial arrangement of vascular bundles. Vascular bundles are radial arrangement. Vascular bundles are in radial arrangement. Xylem and phloem are in different radii. Now, the vascular bundles are here again very scattered and they are in a very patchy form. Vascular bundles are very in a patchy form here. They are in a patchy form here. Now, in the case of roots, I also mentioned, is it exarch or is it endark? Remember students, always in the case of roots, it's always exarch condition. It's always exarch condition. What is the meaning of exarch, sir? In the case of exarch, the X is what? Your protoxylem is towards the exterior side. Here, can you see? Where see? Protoxylem is here. Protoxylem is Here, metaxylem is here. This is metaxylem. This is protoxylem. Protoxylem is towards the outside. If protoxylem is towards the outside, it's called as exarch. Done. Protoxylem towards the outside, it's called as exarch condition. Now, how many vascular bundles can you see here? In case of your dicot root, there are around 2 to 4 vascular bundles. 2 to 4 vascular bundles can be seen here. 2 to 4 vascular bundles. That is called as tetrarch and diarch condition. Diarch and tetrarch condition of vascular bundles. That is diarch and tetrarch. Diarch as well as tetrarch. Now, last point here is between the xylem and phloem. Between the xylem and phloem, there are some type of tissues. Between the xylem and phloem, there are certain tissues called as conjunctive tissue. Between xylem and phloem, between your xylem and phloem, there is something called as your conjunctive tissue. Conjunctive tissue. There is something called as what? Conjunctive tissue. That is the entire vascular bundle, details written step by step. Now, what about pith? Don't forget, I didn't forget about your pith. In the case of your dicot root, the pith is actually very, 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 very small. The pith is so small, NCERT uses a word called as inconspicuous. The pith here is something called as what? Inconspicuous here. The pith is what? Inconspicuous. The pith here is something called as inconspicuous, very, very tiny pith. Now, let me tell you one more line here. Let me tell you one more point. What is a stele here? What is a stele? Stele is nothing but your pericycle plus vascular bundles plus the pith region or the pith region. That is your stele. That is the stele. This is the pith here in the center. It is actually very tiny. This is they have shown is big, but it's actually very tiny and inconspicuous. What is stele? Pericycle plus vascular bundles plus pith is your stele. Here. That is the end of your dicot root. Now let's listen to me very carefully. In 2 minutes, I will summarize the entire dicot root. Listen to me very carefully now. From starting, I'll summarize the entire thing. Listen to me very, very carefully. So what do we have here? First, we have your epidermis. Epidermis is single layer. It's called as epiblema. It is made of parenchyma. It has root hair. Connective cuticle is absent. Stomata is absent. Intercellular space is completely absent in epidermis. Then we have your cortex. Cortex is made up of parenchyma. Many or multi-layer cortex. Intercellular space is present. Now what about endodermis? The endodermis is a single layer part of your cortex made up of parenchyma, acts as a checkpoint. Intercellular space is completely absent. The cells here are barrel-shaped cells with suberine deposition, which makes the Casparian strips. Then we have the pericycle. Pericycle is few layer and made up of parenchyma. Two main functions: lateral roots, secondary growth, endogenesis origin. Then we have your vascular bundles, radial, alternate arrangement, patchy, exarch, 2 to 4 vascular bundles. That is called as your diarch condition or tetrarch condition. Between the xylem and phloem, we have the conjunctive tissue. Pith is very small, inconspicuous. Stele is something but your pericycle plus vascular bundles plus pith. That is your stele. That is your dicot stem diagram. The entire diagram is decoded. For you. Now, I want each and every one of you to do one thing. Please download the notes and make these notes in your notebook, all of you. Will you do that? Download the notes, make the notes in your NCERT just like this or in your notebook. Okay. See, NCERT lines. NCERT also says this. What is it NCERT says? the transverse section of your sunflower root, dicot root, internal tissues are following. The outermost layer is called as epiblema. Many cells of the epiblema protrude in the form of your unicellular root hair. The cortex consists of your several layer. I told you, cortex is many layer, thin-walled parenchyma cells. I told you, wrote the point, with intercellular spaces present. The innermost layer of your cortex is called as your endodermis, which is part of your cortex innermost layer. It comprises of your single layer barrel-shaped cells without any intercellular space. Barrel-shaped cells, no space. The tangentially, I told you, tangentially as well as radially, radial walls of the endodermal cells have a depositions of water impermeable waxy material called as suberin. Suberin deposition will make sure water does not enter. But if students, if the suberin was completely surrounded, water would not pass through. That is why NCERT is telling you depositions of suberin is only where? Tangentially and radially, not throughout the barrel-shaped cells, not throughout the endodermis, only and only tangentially and radially, which form the Casparian strips. Next to endodermis is few layers of thick-walled parenchyma cells called as pericycle, initials of lateral roots as well as vascular cambium. During the secondary growth takes place with the help of pericycle. Now the pith is very small, I told you, small pith or inconspicuous, very inconspicuous pith. Growth takes place in these cells, the pith is done. Parenchyma cells, which lies between your xylem and phloem, is called as your conjunctive tissue. Between xylem and phloem, it is conjunctive tissue. There are usually 2 or 2 to 4, right? 2 to 4, I told you, 2 to 4 diarch or tetrarch condition. Phloem patches. Later, the cambium ring develops between the xylem and phloem. All tissues on the inside of the endodermis, such as your pericycle plus vascular bundle plus the pith, is called as stele. That is called as your stele. Every line of NCERT, with the help of diagram, I made sure you write point wise. Okay, I don't think we missed even a single line of your NCERT. I feel we didn't even miss a single line of NCERT is not missed. Every single line is covered. Okay, that's how I want every single student of mine to understand every single chapter. Okay, even everyone should love this chapter. This is how the chapter is. Amazing, all of you are. Now let's understand monocot root. Very simple. Monocot root and dicot root are almost same. Monocot root and dicot root are almost same, but there are small changes. What are the small changes? See, I'll tell you first. Normally, in the case of your monocot root, epidermis, cortex and pericycle. Pericycle is almost same in the case of your dicot and monocot. But the first difference comes in your vascular bundles. The first difference comes in your vascular bundles. Now what is the difference here? In the case of your vascular bundles, in your dicot root, did I write 2 to 4 diarch to tetrarch condition? But in the case here, how many vascular bundles are here? 1, 2, 3, 4, 5, 6, 7, 8, I guess. That is called as your polyarch condition. That is called as what? Polyarch condition. Here in the case of your monocot root, the vascular bundles are in your polyarch condition. They are in your polyarch condition. Clear? Now what about pith here, sir? Pith. In the case of your monocot root, the pith is actually very large. Pith is very large here. Large pith. Very large pith. Can I also tell it is conspicuous now? It is something called as conspicuous and well developed. It is conspicuous as well as well developed. Well developed here. Conspicuous as well as well developed. Now here, since it is monocot root, the cambium will be open or close. You tell me. I think I forgot to mention here. Forgot to mention here. It is open. It is open. Vascular bundle will be present in the case of your dicot root which shows secondary growth. Okay. Here in the case of here, vascular bundles will be closed because monocots do not show secondary growth. So vascular bundles are closed here. Now what about the arrangement? Arrangement is again radial arrangement because it is root, and also X-arch condition is seen because it is root. X-arch condition is seen in the case of your root. Secondary growth is absent. Secondary growth is absent here. That is why you don't see long roots here. In the case of monocots, we have fibrous roots, right? Fibrous root. That is why secondary growth is absent here. Clear? Is every point clear to all of you? No KMBM, no secondary growth. Exactly. NCERT lines. Let's see the NCERT lines. Monocotyledonous root. The anatomy of monocot is similar to your dicot in many respects. It has epidermis, cortex and endodermis, pericycle, vascular bundles and pith. Same. As compared to dicot root which has fewer vascular bundles, there are usually more than 6. Polyarch condition. First point I told you. Xylem bundles in the monocot. The pith is large and well developed. I made you write down. Monocotyledon roots do not undergo any secondary growth. No cambium, no secondary growth. No secondary growth. Yes. Secondary growth. Then we have your, that's all. That is your NCERT says. Your NCERT says that much only. But here you need to notice here is, the endodermis here is 2 layer. Can you see 1 layer, 2 layer? Diagram you see. Diagram it shows 1 and 2 layer. So your endodermis is 2 layer. But it is not present in your NCERT. They will not ask you this. They will not ask you this, but endodermis is 2 layered or few layered. It is not given in your NCERT, but it is important. It is not given anywhere. But actually, in some cases, see, I told you there are always exceptions, right? There's always exceptions. In the case of your root also, in some cases it is few layer. In some cases it is single layer. But please learn it as single layer. But in the case of, you can see diagram, it shows 2 layer. So there are 2 layers or few layers. But NCERT is not mentioning that, so you don't have to worry about it. NCERT is not mentioning is, so you don't have to worry about it. Okay. So NCERT is only giving this paragraph, only worry about this paragraph. That's what I'm asking. Nothing extra, nothing less. Okay. Now, let's understand dicot stem now. Can we start dicot stem? Yes, crazy. Secondary growth is completely deleted. Secondary growth will not be asked. Okay. Secondary growth is completely absent. Cool. Diagram is important. Let me teach you with the help of diagram again. Can we start? So, any of you students who was watching till now, who was started in the starting with me, some of the students I can see who started, Pandu is there, Jayaprakash is there. Some of you started with me at the end of the starting of the class, or approximately 3 hours before. Some of you are still here with me. If you're understanding, right, please hit the like button, subscribe to the channel, and also do not forget to today's video. I want each and every one of you comment down. If you have any doubt, please comment down in today's video. Let me know how the session was. Okay. Water is missing here. No, I have water. Oh, I have water. I forgot. I thought I didn't have water. Thank you so much. All of you drink some water. Do any of you have any doubt in the case of your dicot root? Ask me right now. Dicot root, any doubt? Ask me right now. If not, I will start with your dicot stem now. Amazing. Yes, MD was also there from starting. Shivani is also there from starting. Now, similarly, again, I will teach you epidermis, then we go to cortex, then we teach your pericycle, endodermis, phloem, then we do your vascular bundles, then we go to your pith. Similar fashion. just like that, one by one, we'll be doing now. Okay. So before, no doubt, amazing, no doubts, we can start with your dicot stem. Now let's start with epidermis. Epidermis. The first one is your epidermis here. Now, epidermis again is made up of your parenchyma cells. Yes. Epidermis is made up of your parenchyma cells. Epidermis is again made up of your parenchyma cells. Now, in the case of your stem, do we have stem hair? And we call the stem hair as trichomes. Yes, the stem hair, can you see? Epidermis has something called as your what? Epidermal hair. Epidermal hair is also called as your what? Trichomes. Yes, they're also called as your trichomes. Trichomes, also called as your epidermal hair. Epidermal hair. Yes, it's called as epidermal hair. Then, in the case of your epidermis, is there intercellular spaces? No, sir. In the case of your epidermis, the intercellular spaces are completely absent. In the case of epidermis, the intercellular spaces are completely absent. Now, students, you tell me in the chat right now, is the cuticle is present? In the case of your stem, the cuticle may be present or may be absent. It can be maybe present or may be absent. Now, what about stomata? Just like your stem, just like your cuticle, stomata may be present in some cases. In some cases, stomata will be absent in the case of your dicot stem. The stomata can be present or stomata can be absent. Now, the last point, the most important point here, your epidermis. Epidermis is single layer. Epidermis is your single layer. Clear, all of you? So, in the case of your epidermis, it is made up of your parenchyma, trichomes or epidermal hair is present, intercellular space is absent, cuticle may be present, may be absent. What about stomata? Stomata may be present, may be absent. Then we have epidermis as a single layer. Epidermis is single layer. Amazing. Epidermis is single layer. Done. Any doubt in epidermis? Ask me right now. Any doubt here? No doubt? Amazing. Next we go to the next one, that is hypodermis. Next, below the epidermis, we have the hypodermis. What do we have? Below the epidermis, we have the hypodermis. Below the epidermis, we have the hypodermis. Now, students, you tell me in the chat. Now, this hypodermis, in the case of your dicot stem, in the case of your petiole, is made up of collenchyma. Hypodermis is made up of what? Collenchyma. It is made up of what? Collenchyma. It is a few layer thick. It is few layer. Can you see? NCERT also mentions collenchyma. NCERT is also mentioning collenchyma. That is your hypodermis. Collenchyma. Now, do you find intercellular space anywhere here? Absolutely not. In the case of your hypodermis, intercellular space are completely absent. Now, what is the function of your collenchyma? All of us know the function of collenchyma or the function of your hypodermis here is mechanical support. Yes, mechanical support. Apart from mechanical support, remember, collenchyma is living or dead? Collenchyma was living. It has chloroplast. Can collenchyma help in do photosynthesis? Yes. It can help in your photosynthesis. It can help in what? Photosynthesis. Those are all the following parts about your hypodermis. Any doubt here? Amazing. No doubt at all. So we have collenchyma, few layer, intercellular space is absent, mechanical support, photosynthesis. Amazing. Now, below the hypodermis, we have the cortex. Now students, listen to me very carefully, each and every one of you. This particular cortex right here is divided into 3 subzones. Cortex is divided into 3 subzones. Cortex is divided into 3 subzones. The first subzone was actually your hypodermis. Hypodermis is actually part of your what? Cortex. Hypodermis is part of your cortex. That is the first subzone. First subzone was your hypodermis itself. Okay. First subzone was your hypodermis. The second one is your cortical. The second one is your cortical layer. Hypodermis, cortical layer, and then we have your complete parenchyma. Complete parenchyma. Can you see? Complete parenchyma. It is complete. Then we have complete parenchyma. Then we have complete parenchyma. If there is parenchyma is present, does it have intercellular spaces? You tell me in the chat right now. Yes, sir. If there is parenchyma, there are intercellular spaces here. Intercellular spaces present. Intercellular spaces present. Now, can you see this parenchyma is not just one layer. 1 layer, 2 layer, 3 layer. So, can I write few layer thick? It is few layer thick. That is your cortex. That is your cortex. Cortex is given 3 subzones: hypodermis, cortical layer, and parenchyma. Intercellular space is present here. It is few layer. It is few layer. Then what do we have? Then we have your endodermis. Then we have your endodermis. Then we have your endodermis. Now students, listen to me very carefully. Again, endodermis is again single layer. But here, endodermis is little special. The endodermis, in the case of your dicot stem, can store certain substances, something like your starch. So there is something called as your starch sheath depositions in the case of your endodermis. In the case of your endodermis, there is depositions of what? Depositions of your starch sheath. In the case of your endodermis, there is depositions of starch sheath. Starch sheath depositions is there. That is the very important point here. Very important point here. Okay, that's where the question can be asked for you. Actually, question, if you ask me, in your dicot stem, this is one of the points where question can be asked. The other point is your collenchyma. That is also very important. Now, let's understand pericycle. There also question can come. Let's understand pericycle. Pericycle. Now, pericycle here, if you notice, the pericycle here, can you see, it is a little wavy in nature. Yes, sir. Pericycle, I can see. Pericycle is actually wavy in nature. Pericycle is what? Wavy in nature. It is wavy in nature. It is very wavy. Now, what is the shape of pericycle? Can you see this? The shape of the pericycle is half moon shape. This is your full moon. This is the full moon. And then we have the half moon. Half moon is called as what? The half moon is called as your semi-lunar. It is called as what? Semi-lunar shape. Semi-lunar. It is in a semi-lunar shape. Now, the biggest question of all. What is pericycle made up of? Pericycle is made up of your sclerenchyma. Very important point. Pericycle is made up of your sclerenchyma. Pericycle is made up of sclerenchyma here. Pericycle is made up of sclerenchyma. Clear? Pericycle is made up of sclerenchyma here. So, pericycle is wavy in nature, semi-lunar in shape, as well as it is made up of sclerenchyma. Understood? Any doubt here? Ask me right now. If you have any doubt, this is the point you ask me. Okay. Endodermis has starch sheet. Pericycle is made up of your wavy in nature, semi-lunar in shape, and then we have your it is made up of sclerenchyma. Okay. Now, the next point I want all of you to understand here is the vascular bundles. Oh, there is no space for vascular bundles. There is no space for vascular bundles. Where do I write? Can I write here only? Okay, let's write here. The small gap I'll write here. Vascular bundles. I want one more. I wanted. One second, students. Let me add one more slide here. No. Let's not add one more slide. It's fine. I'll write here in the corner somewhere. In the corner only, we'll write here. The vascular bundles here. The vascular bundles here are very important. The vascular bundles here is what? Can you see there in a ring arrangement? Yes, sir. Vascular bundles are here in a ring arrangement here. Vascular bundles are in a complete ring arrangement. Vascular bundles are in a ring arrangement. Ring arrangement. Yes, vascular bundles are in ring arrangement. Now I told you, in the case of your root, it is conjoint. But in the case of stem, can you see this? This is your, where is the labeling? Labeling is not given here. See, xylem and phloem. Here can you see? Protoxylem, metaxylem and phloem. Your phloem and xylem, are they present on the same radii? Yes, sir, they are present on the same radii. So can I call them? So can I tell here it is conjoint. Conjoint. Now, xylem is present towards the pith. If xylem is present towards the pith, it is called as collateral. Yes, here we learnt. We learnt here, remember? We learnt here. Can you see? If xylem is present towards the center, it is called as collateral. It is called as what? It is called as your collateral. Conjoint, collateral, and also are they open or closed? In the case of your dicot stem, secondary growth actually happens. That's why they are open. Can you see here? In the case of between your xylem and phloem, there is vascular. In your xylem, there is cambium is present. That's why they are open. That's why they are open. Now here, xylem, it is open. Clear? It is open. Next, listen to me very carefully. If protoxylem is present towards the pith, it is called as your endark, inside. Endo means inside. If protoxylem is inside, it's called as endark. If protoxylem is present towards exarch, outside, that is called as exarch. This is endark condition. It is endark condition. Endarch condition. Endarch condition. I hope those points are clear to all of you. Now let me understand the pith. Let's understand pith here. Let's understand pith. Pith. The pith here is actually very large. The pith is very large here. Can you see? A large central pith. A large central pith. Large central pith. And this particular pith is made up of your parenchyma. Can you see green color? The pith is made up of your parenchyma. Do you see parenchyma? Clear? Now, listen to me very carefully. Listen to me very, very carefully, Rehna. That is, in the case of your pith, between your xylem and phloem, from your pith region, between the xylem and phloem, here, between xylem and phloem, what runs? Between the xylem and phloem, there are medullary rays. From your pith region, from the medulla region, between your xylem and phloem, there are medullary rays. The same medullary rays later gets converted to your interfascicular cambium. Becomes interfascicular cambium. Clear? So from the pith we have rays coming out. It's called as medullary rays. Clear? Medullary rays. That is from the medullary rays from the pith. Now these medullary rays are made up of your parenchyma. They are made up of your parenchyma. Medullary rays are made up of what? Parenchyma. Students, Sumaya, all of you students, if you all this information can be a little too much, I know. But if you read NCERT, for all of this, it can become very confusing. because diagram is given some side, information is given some other side. When you learn with the help of diagram, you will understand it better. You will understand this better. Clear? That's all. Amazing. So intra or inter, someone is asking me intra or inter. Lilac flu. I hope that is not is that your real name, or let me tell you your real name. Remember, your intra is primary in origin. Intra is primary in origin. Now I just told you, medullary race gets converted to something. Medullary race is getting de-differentiated. Means it has to be your inter. Okay, it has to be inter. Intra is primary meristrum. Inter is your secondary meristrum. Remember that? Amazing. Let's quickly revise with the help of NCERT lines. Let's revise with the help of NCERT lines here. What does NCERT line says? NCERT line says very simple. The transposition of your typical young dicot stem shows the epidermis, already mentioned, is the outermost layer, productive layer of the stem. The covered with a tiny hair of cuticle. Yes, there is a layer of cuticle on the epidermis. It may bear trichome and a few stomata. Stomata may will be present, it may will be absent. Then we have your cells arranged in multiple layers between the epidermis and pericycle constitute the cortex. Cortex is divided into 3 layers. It consists of 3 subzones. The outer hypodermis, I told you, hypodermis is part of your 1 layer. Hypodermis consists of your few layer collenchyma, mentioned already. Hypodermis is made up of your collenchyma. Just below the epidermis is your hypodermis, which provides mechanical strength, mentioned already, to young stem. The cortical layers, told you next, cortical layers are below your hypodermis, consist of your rounded, thin-walled parenchymal cells with conspicuous interstellar space. Parenchyma has interstellar spaces here. The innermost layer of your cortex is called as your endodermis, third layer of your cortex. Then we have the cells of the endodermis are rich in starch sheath. That is, starch sheath is present. The pericycle is present in the inner side of your endodermis and above the phloem in the semilunar patch. Yes, semilunar patch. Then it is made up of sclerenchyma, mentioned already. In between the vascular bundles, there is a few layers of radially placed parenchymal cells which constitute the medullary race. So remember, medullary race is made up of parenchyma between the vascular bundles. See, I told you what is present between vascular bundles. Between the vascular bundles, we have the interfascicular cambium, right? And between the xylem and phloem, it is intra. Okay, a large number of vascular bundles are arranged in a ring. Ring arrangement, I told you. Ring arrangement of vascular bundles as a character of your dicot stem. Each vascular bundle is conjoined. What is the meaning of conjoined? The meaning of conjoined is they are present on your same radii, xylem and phloem. Open cambium is present and with endark protoxylem. Protoxylem towards the inside, it is endark condition. A large number of round parenchymal cells with the large interstellar spaces occupy the center portion of the stem constitute the pith. Large cells with interstellar space. I think I forgot to mention that. ICS is present. In case of your pith, ICS, interstellar space is present. Interstellar space is present in the case of your pith. Clear? Yes, it is present only in the case of your monocots. See, remember, sclerenchyma is mentioned in the case of monocots, yes, as well as medullary race. So dicot stem is over. Any doubt in dicot stem? Any doubt in NCERT lines? Any doubts in the NCERT lines? Nothing. Can we start monocot stem? Dinner break? No. We'll finish everything and then I'll go home and then you eat dinner, I eat dinner at my home. Okay. Because my house is around 22 kilometers from here. I have to drive and go for 22 kilometers, then my dinner. Okay. Amazing. Can we start with your monocot stem now? Monocot stem is very simple. Monocot stem is very, very simple. Again, we have the epidermis here. Again, we have the dermis here. Now, again we have epidermis. So epidermis is very simple, same epidermis, almost the same. Epidermis is very simple here. Then we have your hypodermis. This is where is important. Hypodermis. Now I told you students, all the future doctors, in the case of your dicot stem, hypodermis was made up of collenchyma. But in the case of your monocot stem, hypodermis is made up of what? Hypodermis is made up of your sclerenchyma. Hypodermis is made up of what? Sclerenchyma. Hypodermis is made up of sclerenchyma. So students, in the case of your monocot stem, what about the ground tissues? In the case of your monocot stem, the ground tissues are completely scattered. See, in the case of dicot, there was a ring arrangement, right? There was a ring arrangement in the case of dicot. But in the case of monocot, vascular bundles are completely scattered. Vascular bundles are completely scattered. Ground tissue, that is, vascular bundles are not different. Ground tissue is not differentiated. Can you see any differentiation of ground tissue? Ground tissue is not differentiated. Completely not differentiated. That is, and it is made up of your parenchyma. It is made up of the parenchyma. Clear? Ground tissue is made up of your parenchyma. Now, what about vascular bundles? What about vascular bundles? I already told you, vascular bundles are, yes, scattered. Vascular bundles are completely scattered. Vascular bundles are completely scattered here. Complete scattering. Can you see? See this? Vascular bundles are everywhere. Throughout the tissue, throughout the monocot stem, vascular bundles can be seen be completely scattered everywhere. Okay. Now, here one most important point I want all of you to understand here is, can you see vascular bundles towards the outside, they are more in number? Yes, sir. Towards the outside, vascular bundles are more in number. As you go towards the center, they are less in number. Yes. Vascular bundles towards the outside are more in number, but they are small size. So write down, vascular bundles towards the outside are, outside there are more vascular bundles. Towards the outside, there are more vascular bundles, but small size. But they are what? Small size. They are very small size. Now, but look at the inside. Inside, the vascular bundles are very large. Inside, the vascular bundles are very large, but less in number. So if you look at inside, that is less vascular bundles, but large size. Very large size. Now, clear? Next point here is the vascular bundles has a covering. Vascular bundle has a covering here. In the case of stem, vascular bundles is completely surrounded. That is, xylem and phloem are completely surrounded by a sheet called as bundle sheet. They're covered by your bundle sheet covering. Bundle sheet covering, and this bundle sheet covering is made up of your sclerenchyma. Most important line. This bundle sheet is made up of your sclerenchyma. Please write down this. Is made up of your sclerenchyma. Very, very important. A question can come here. Now I'll tell you what is even more important. Even more important line is I'll tell you. Even more important here is the phloem parenchyma is absent here. Phloem parenchyma is absent. We learned, remember, phloem vessels, right? We learned about your phloem, right? Companion cells, sieve elements, phloem fibers, phloem parenchyma. Here, phloem parenchyma is absent. Phloem parenchyma is absent. Clear? Now, vascular bundles here. Vascular bundles are here. Vascular bundles are here conjoined. Why conjoined? Because this is the case of your stem. What is the meaning of conjoint? The meaning of conjoint here is the xylem and phloem are present on the same radii. And the vascular bundles are here closed. What is the meaning of closed vascular bundles? The meaning of closed here is the cambium is completely absent here. The cambium is completely absent here. Cambium is completely absent here. Clear? The last point here. The last point here is inside the cambium, can you see this blue color structure? Blue color structure here, can you see? I can see, sir. What are those? Those are water cavities. That is inside the vascular bundle. Inside the vascular bundle, there are water containing cavities. Water containing cavities. Inside the vascular bundle, there are water containing cavities. Clear? That is your monocot stem. So I'll tell you, revise one more time. In monocot stem, we have the epidermis, which is a common epidermis. Then we have your hypodermis, made up of your sclerenchyma. Very important, I told you. Then we have ground tissue, not differentiated at all. Ground tissue is completely not differentiated at all. That is, it is made up of your parenchyma. Then we have vascular bundles. Vascular bundles are completely scattered here. Everywhere, vascular bundles are completely scattered. Then we have vascular bundles towards the outside, are large in number. They are more in number and very small. But as you go towards the center, the number is decreasing of vascular bundles, but the size is increasing. Now, phloem parenchyma is absent here. Very important. In the case of your monocot stem, the phloem parenchyma is completely absent. Now, in the case of your vascular bundles, there is an extra covering on top of your vascular bundles that is called as bundle sheet, and the bundle sheet is made up of your sclerenchyma. Very important point. Then we have vascular bundles are conjoined, that is one after another. Xylem and phloem is present. It is closed. The cambium is absent. The cambium is absent. Then there is water containing cavity here. There is water containing cavity here. Points are clear. I hope every one of you are learning anatomy today, understanding anatomy, but please make these notes on your own. Download my PDF. Go to Telegram channel. Download this PDF, write down the notes from the PDF also. Okay. Now, NCERT lines. See, the monocot stem has sclerenchymatous hypodermis. I told you, write down the point here. Yes, make sure you write down the point. A large number of your scattered vascular bundles. Yes, vascular bundles are completely scattered, each surrounded by a sclerenchymatous bundle sheet. I made you write that point. And a large conspicuous parenchymatous ground tissue. Parenchymatous ground tissue is there. The vascular bundles are conjoined and closed. Done. The peripheral vascular bundles, means perivascular bundles towards the outside, are generally smaller. Yes, but large, more in number than centrally located ones. The phloem parenchyma is absent here. Important line. The phloem parenchyma is completely absent. And water containing cavities are present within the vascular bundles. Clear? That is the monocot stem. Clear? Yes, there is pith, but it's not mentioned in your NCERT. I don't know why. See, the pith is not actually, the reason the pith is not very visible here is because the ground tissue is so scattered. The ground tissue is so not differentiated at all. The pith is actually not seen at all. It is very inconspicuous. No pith is not seen at all because your ground tissue is only scattered. Where will the pith be formed? Right? Where will the pith will be formed? The pith is only formed when the vascular bundles are arranged properly. Even the vascular bundles are scattered, so pith is also not formed. Okay? Amazing, all of you are staying for such a long time. Now, the last part of the chapter is your leaf. Last part of the chapter is your leaf now. We have come to the end part of your chapter, that is leaf. Can we start? This is the last 10-15 minutes and I will clear the entire, done and dusted, the anatomy of flowering plants. 14 marks in your pocket. All you need to do is revise the chapter one more time, and you will be done and dusted. Please revise. Okay. Now, Last part of the chapter is your leaf. Can we start leaf? Can we start? Amazing. So I can't believe some of the students are still there. Now someone was asking me about PYQ. Before I teach you about leaf, do you want to solve PYQs? Do you want to solve more PYQs? Akansha, Pandu, Somaya, Crazy. Do you want to solve more PYQs on this chapter? Link is in the description. I made sure you solve some PYQs on live. But more PYQs, if you want to solve, a PDF is available in the description, students. Go on, please click on the PDF, download the PDF, and also solve those questions. Then only, truly, it will be done and dusted for you. And students, in your NEET examination, there is just hardly 100 days left, right? Just 100 days left. For this next 100 days, you will stay with me on the channel. Yes, after 100 days, you will go to your medical college. I will be here teaching the next group of students, the next future doctors. So I want the next 100 days to be the best 100 days for you. So for that to happen, I want each and every one of you to be consistent on the channel. I know I started the video with 40-50 people watching. Now we just have around 25 people, 26 people. But this is what I do not mind, these 25 people, because when I started teaching this chapter for the first time on YouTube, guess how many people were there? When I taught this chapter for the first time on YouTube, there were only 2 people or 3 people. Now we are 25 people here, 25 students. So that is a growth from my side. That is a growth from your side also. So next 100 days, your time on this channel, we'll make sure it's going to be fruitful. So please stick with us for the next 100 days and your entire syllabus will be over with PDF of your NCERT questions, as well as your PYQ. Everything is given. Please go on. Just all you do is click on the link, download the PDF, make your own notes, get into your dream medical college. Okay? Get into your dream medical college, all of you. So let's start the last part of the chapter, that is leaf. So all of us know leaf is nothing but a photosynthetic. It is a photosynthetic organ, right? It is a photosynthetic organ. If I draw a typical leaf, can I draw a typical leaf here? This is your typical leaf. Yes, a typical leaf here. Now this particular leaf, this part is called as your lamina. Yes, this is your lamina. This is your midrib. This is your midrib. Now on this particular midrib is also your largest vein. Yes, it is also the largest vein. On this veins, do you see certain veinlets coming out? Yes, sir, we see certain veinlets coming out. Now, I will teach you one particular words here, 2-3 words. 2-3 words. What are the 2-3 words here? Your particular leaf has 2 layers, 2 surfaces. That is, it has something called as your upper epidermis. Upper epidermis. Yes, it also has something called as your lower epidermis. This is your lower epidermis. Lower epi. One second, students. Lower epidermis. Lower epidermis. Yes. Now I'll teach you some other words for it. Please write down the few words. Your epidermis is also called as upper epidermis is also called as your ventral side. Also called as your ventral side. It is also called as what? It is also called as adaxial side. It is also called as your adaxial side. It is also called as what? Adaxial side. Now what about lower epidermis? Lower epidermis is also called as your dorsal side. Dorsal side. It is also called as abaxial side. Backside means lower. It's called as abaxial side. It is called as what? Abaxial side. Abaxial side. Now students, listen to me very carefully. Listen to me very, very carefully, okay? Now this is your midrib. Now students, listen to me. On this midrib, there are vascular bundles. On your veins, also there are vascular bundles. But remember this one word I'll teach you now. In the case of your dicot, we have variable number of vascular bundles depending upon whether they are on midrib or veinlets. Because on your midrib region, there are more vascular bundles. On your midrib region, vascular bundles are more. Because in the case of your dicot, there is something called as variable vascular bundles. Variable vascular bundles. On your midrib, on your dicot leaf, we have something called as variable vascular bundles. And this is the midrib where vascular bundles are very high. And this is a typical structure of your dicot leaf. Typical structure of your dicot leaf. But sir, what about in monocot? Students, all of you know, in the case of your monocot, we have the parallel venation. Yes, this is your reticulate venation. This is your reticulate venation. In the case of your monocots, vascular bundles are actually fixed. Vascular bundles are actually fixed. Vascular bundles are fixed in number. Fixed in monocots. In the case of dicots, in the midrib region, there are more vascular bundles. But in the case of veins and veinlets, less vascular bundles. So in the case of your dicots, variable vascular bundles. In the case of your monocots, it is variable. In the case of your monocots, it is fixed vascular bundles. Fixed vascular bundles in the case of your monocots. Now again, let's look at the diagram. See, I told you, adaxial side. Yes, there is something called as your adaxial side, upper side, abaxial side, lower side. Two different types of epidermis, upper epidermis as well as lower epidermis. Now students, listen to me very carefully. In your lower epidermis, there is more stomata. Why, sir? Why is there more stomata in case of your dicot leaf? If you notice a typical dicot leaf, it looks like this. This side, there is more sunlight. This side, there is more sunlight. If the more stomata is present on the bottom, can it reduce the water loss? Yes, because stomata can close and more water loss is prevented. Water loss is like sunlight is not hitting the stomata. So in the case of your dicot leaf, it looks like this. Stomata is present in the lower side more. Okay, lower side more. Now, what about the cortex? Students, in the case of your dicot leaf, cortex is made up of your parenchyma. It is made up of what? Parenchyma. Now, parenchyma here is present in a type of cell called as your mesophyll cells. We have something called as your mesophyll cells, which is divided into your two types. Mesophyll cells is divided into your two types, that is your palisade cells as well as your spongy mesophyll cells. Mesophyll cells are two types in the case of your dicot leaf, that is your palisade cells as well as spongy mesophyll cells. Listen to me very carefully. Your palisade cells are arranged in a vertical manner. Yes, they are arranged in a vertical manner which are parallel to one another. They are arranged in a vertical manner, but they are parallel to one another. Vertical manner, parallel to one another. But your spongy parenchyma are arranged in a, they are much more cylindrical in nature. They're much more spherical in nature. Spherical or oval in nature with the presence of air cavity. Air cavity is present in your lower side more. Lower side more, we have more air cavity. That is in the case of your dicot leaf. In the case of your dicot leaf. Now here we have your xylem and phloem. Yes, we have xylem and phloem. The vascular bundle is covered by a bundle sheet. Yes, it is covered by a bundle sheet. In the case of your dicot leaf, this bundle sheet is made up of your parenchyma. Bundle sheet is made up of parenchyma. Now students, on the epidermis, On the epidermis, do you find cuticle? Obviously, sir, this is a leaf. What are you talking? Yes, sir, on the leaf, we have cuticle here. We have the cuticle presence here. Cuticle is present here. More stomata on the lower side. We have cortex is divided into two types. Palisade, spongy. Yes, air cavity present on the lower side. More stomata present lower side. More cuticle present on the upper side, because this upper side is exposed to sunlight. So cuticle is present on the upper side more. That's all. Clear? See the NCERT lines. NCERT line says your dicot leaf is called as what? Dorsiventral, because it has a dorsal side and a ventral side. Remember, dorsiventral dicotyledonous leaf. The ventral section of your dorsiventral leaf throughout the lamina shows three major parts. That is epidermis, I told you, mesophyll, as well as vascular system, obviously. Then we have here the epidermis, which covers both upper surface, that is called as what? Adaxial epidermis, I told you, adaxial, abaxial, and lower surface called as your abaxial. The leaf has conspicuous cuticle. Obviously, it's a leaf. If there is a leaf, there is a conspicuous cuticle. The abaxial, that is lower side, generally bears more stomata, already mentioned you, than the adaxial. The later may even lack stomata, that is your upper side even may lack stomata sometimes. Okay, the tissue between your upper and lower epidermis is your mesophyll. Mesophyll is again of two types, spongy as well as palisade. The mesophylls which possess chloroplast and carry out photosynthesis is made up of your parenchyma. Two types, palisade parenchyma as well as your spongy parenchyma. Clear? The adaxial placed palisade parenchyma is elongated cells. Yes, they are elongated cells, but they are parallel to one another. They are arranged vertically. I told you, vertically and parallel to each other. They are arranged vertically. They are arranged vertically but parallel to each other. Diagram is very important. One another. The oval or round, I told you, oval or spherical or round, right? Loosely arranged spongy parenchyma. Yes, spongy parenchyma is situated in the below the palisade. Below the palisade, we have the spongy parenchyma and extend to the lower epidermis. A numerous number of space or air cavities present between the cells. Then we have vascular system. Vascular system includes vascular bundles, which can be seen in the case of your veins and vein. I told you, vascular bundles are present in the veins and midrib. The veins may vary in thickness in reticulate venation. In dicot, the vascular bundles are surrounded by a layer of thick-walled bundle sheet that is made up of your parenchyma. Look at the figure. That is already seen. Then we have monocot leaf. Again, monocot leaf is again divided into your upper epidermis and lower epidermis. Monocot leaf is again divided into your upper epidermis as well as your lower epidermis. But here the mesophyll cell is not divided into your palisade or spongy. Then we have something called as your substomatal cavity here. And we have stomata again. Stomata again here in the case of your monocots. Listen to me very carefully. Listen to me very, very carefully. This is a monocot leaf. Monocot leaf will be like this something. Yes, have you seen grasses? Have you seen grasses? They look something like this. Right? Monocot leaf. So in the case of monocot leaf, the sunlight is reaching both the sides more all the time. This is your leafing sheath base. Monocot leaf, stomata are almost equal. Stomata are almost equal on both the sides in the case of your monocot leaf. Clear? That is the anatomy of isobilateral, they call it as isobilateral. Iso means same, bilateral means two sides are same here almost. Similar, that is the dorsiventral leaf. It shows the following characters. What are the following characters? The stomata are present on both surfaces. I just told you, stomata are present on both the surfaces equally of epidermis, and mesophyll cell is not differentiated into your palisade as well as spongy parenchyma. Now, this is where the question will come from. In grasses, a certain adaxial epidermal cells. Adaxial is what? Adaxial is your top cells. Some of the top cells in the case of your monocot leaf can become colorless. Right? They can become large. They can become large, empty, colorless. Some of the epidermal cells can become modified into large, colorless cells called as bulliform cells. These are called as bulliform cells. When the bulliform cells in the leaves have absorbed water, they are turgid. So, example, this is your bulliform cell. If it has absorbed water, it looks like this, turgid, very turgid. Bulliform cell has absorbed water. Now, the leaf surface is exposed. Yes, leaf surface, complete leaf surface is exposed now. Sunlight will come and fall. Leaf surface is exposed. Now, when the flaccid, due to the water stress, whenever there is water stress, all the water is gone. Yes, all the water is gone. Now, they make leaves curl inwards and minimize the water loss. See here, whenever there is water loss, they will curve like this. They will curve like this. Have you seen certain grasses? Have you seen certain grasses? The leaf of the grass is like this. In the case of your rice leaf, right? Rice leaf, jawar leaf, the leaf will curl like this little bit. The leaf will show curling. That is because of the bulliform cells. Whenever there is less water, the leaf will curve like this little bit. Not like this. It will curve, leaves will curve like this. Clear? The parallel venation in monocot leaves reflects the near similar size of the vascular bundles. I already mentioned, in your vascular bundles are almost same in number. Also always same in number, vascular bundles, except in the main vein, as seen in the case of your vertical section of the leaf. So this is your homework question. I think I already told you the answer. I think I already mentioned this question. What is the answer? The large empty color cells is your bulliform cells, not homework question. Not your homework question. That is the end of the chapter actually. Is the thumbnail wrong? Students, is the thumbnail wrong? All of you tell me in the chat right now. Is the thumbnail wrong? That is the end of the chapter. That is the end of the chapter now. Clear? Is every point clear to you? Is every point clear to all of you? How was today's session? Let me know right now. Let me see here only. If every point is clear to you, let me know right now here. Only 22 likes, sir. What is this? I can only see 22. I think it's not refreshed. Yes, I think it is not refreshed. That's why it's showing like this. Okay. So students, I want each and every one of you. So this has been a very long journey for me also. I started with 2 live watching. In that 2 live watching, one used to be mine only. Okay. In that 2 live watching, one used to be mine only. Now I've come certain path. I've traveled some path. Students, it's your job to let me know in today's session in comments. Please let me know if you have any doubts. Any doubts you can ask me in your today's comments. Any doubt you can ask me. I will personally reply. I will personally reply to every message if you have any doubt. Also, let me know how was session today. Now, other thing I want all of you to do is, here, go to the description. Can you see here? Can you see? Oops, almost dropped the bottle. If you see here, Telegram channel, first link, you can join the Telegram channel. So to join the Telegram channel, there are more than your 4.6 students, 1000 students are there. Join the Telegram channel, everything is available there. Then we have your NCERT questions, click on it. You get your instructions, start test. Do the start test, that's all. See here, interface of KVM dollars built from the soil. Direct test is there. You don't even have to do anything. In total, there are 19 questions here. Can you see? 19 questions, direct test is starting. You have a time limit also, 30 minutes. Are you seeing that? Anatomy of NEET PYQ, everything for free. Then you see here, go to here, last 7-year PYQ on the chapter, get asked 500 questions. Famous books. See here, you can get all the books here. Clear? Then last 10-year paper, click on it, biology. Just do the login here. Last 10-year questions are available for free completely. You don't have to pay a single rupee. You don't have to buy any book. No book you have to buy in the world. Just go here. You'll get the questions. So this is the best thing is this one. Best thing is this one here. Can you see? Test. So go check your knowledge here only. You can check the knowledge here only. You'll get a scorecard for free completely. Okay. See here. For your chemistry 10-year papers, every single thing is there. You don't have to do anything. You don't have to go anywhere. You don't have to go anywhere. Every single thing is there. So students, my job is done now. My job is done and dusted. I have taught you the entire chapter, done and dusted. Now your job is to go. First thing is give the test here. NCERT questions. Last 7 year PYQ. Download the questions and do it. And go to the Telegram channel. On this Telegram channel right here, the notes. That is the most important thing. I made sure you write so much today, right? All the notes will be provided on this Telegram channel. Your Telegram channel, you can go to, I don't think it will open here, but you can do it in your book, you can do it in your phone. Telegram channel, please go download the notes. That is the end of today's session. Okay, that is the end of today's session. Done and dusted. Students, let me know in today's comment section. How was the session? That is very important. I want to know the feedback. Feedback is the most important because sometimes I might be speaking a little fast. Sometimes some of the concepts you didn't understand. So let me know in the comment section, how was the feedback? How was today's class? That is very important. Okay. So with that, can we stop today's class? I will see you. One more thing. Please let me know also which is the next chapter you want. Can I see here? Which is the next chapter you want? Anyone who can tell me? Which is the next chapter you want? Tell me first. I was thinking, can I teach you human reproduction? Can I teach human reproduction? Can I teach trust me, I can teach human reproduction very well. If you want, I can teach you human reproduction. If you want, I can also teach you biomolecules. Which is it? Biomolecules or human reproduction? Tell me first. Live chat only, no? Biomolecules. Biomolecules or human reproduction? Tell me first. Because I will teach you biomolecules, every line of NCERT, every single formula, every single reaction, every single molecule I'll be teaching you. Every single experiment also I'll be teaching you. What is the meaning of supernate? What is the meaning of your, retentate? Supernate, retentate. What is your type of biomolecules based on experiments, right? Biomolecules, human reproduction, biomolecules. Okay, anyway, let me know, Ravindrani, all of you, let me know in the comment section, which is the next chapter also you need from my side. I can teach you biomolecules or I can teach you human reproduction. Very important, both are. Okay, let me leave a poll based on what is more requirement. I will teach that chapter. Trust me, I will make it very easy for all of you. Okay then, take care. Good night, all of you.